almost 100 in the approval state, then meaning running phase 3 trials. There's a dominance of gene-modified cell therapy and about 400. That's a clear increase, more than 100 more than last year, within 1 year. There's gene therapy, also almost 400. In vivo gene therapy is also an increase. We have a dramatic increase, even if the number is low, in tissue engineering, by doubling the number of clinical trials compared to 2018. Interestingly, there's a reduction of clinical trials in somatic cell therapy, 100 less than last year. And you see also there's a lot of companies around. It's not only big pharma, it's a lot of small companies coming up, technology providers and developer. And you see in summary about 1,000 companies worldwide with a dominance in US, but also in Europe and Asia. And it's interesting, for example, if you speak about Israel, Israel have almost the same number of companies in this field as Germany, but it's only 10% of population. That's showing how important this topic is taken there. If you look for clinical indication, there's a broad spectrum. Of course, there's a dominance in oncology, about 50 to 60%, depending on the region, of clinical trials are in oncology. But you see practically all other clinical indications are also in the target, immune diseases, cardiovascular diseases, and so on. Whereas Europe and Israel together, where we are in this direction. If you see here in the phase 3 studies, about 50% of studies are run in Europe and Israel. But if you go back to the phase 1, you see only 15%. What does it mean? That means obviously there's much more activity now in US and China than in Europe, because in the new pipeline is much more activity there. And you see it also if you look for the different indications or different tools. In gene therapy, 32, 20%, almost 20, 36%, Europe and Israel in comparison to the whole world. The good thing is if you look for global financing and investments in this field for acquisition, for venture capital and so on, is worldwide 2 billion last year. And Europe and Israel together have 1.3 billion, which is really a significant increase. Interestingly, worldwide, it was a little bit drop in the last 12 months, but you have to take into account that in 2017-18 was a dramatic increase. I think the problem is we have a lot of clinical trials, but we have also many, many roadblocks which prevent no more than a good dozen advanced therapies from being approved to date. And Europe is at 11. And you see the problem is from the beginning of the mission-driven basic science to the implementation along a lot of roadblocks. And this is a problem that each of the products currently approved need about 20 years, which means it's a long and costly added value chain, and the final product have a high cost of goods. And the reason for this is clear: it's a disruptive process, a disruptive innovation, practically at all levels of the innovation or the added value chain of the drug development, from the drug discovery, the raw material, manufacturing, preclinical models, mode of action, clinical trial design, PK, PD, scaling up, supply chain, health economy, and last but not least, ethics and complex data, which are generated here by these complex drugs, and which change, of course, also the business models. And that is the reason that we need completely new structures in this field. I think there's a clear high need for targeted effort and sustainable large-scale research initiative in Europe. And this was our motivation to form 1.5 years ago Restore, or a little bit more than 1 year now, to motivate Europe to have a clear perspective in this direction. And what is the need for this? I think on one hand, of course, transpersonality is not longer that it can be done by simple groups or in one pharma company. You need technology, definitely a lot of technologies. And of course, translational structures and academic hubs, including refined translation. That means we need academia, industry partnerships at a new level, and really an interaction and not a sequential collaboration. And very important is the alliances with the patients and their representatives. And who is behind the Restore community? We have this original core team, which is a mixture of academic centers, translational centers, biotech companies, and non-for-profit organizations from different European countries and Israel. And I think it's a very active group, but we could not be successful without all the supporters. At the moment, we have more than 300 supporters who signed the interest for this and help us in developing the roadmap. From 21 countries, of course, member states, associated countries, but also US, Canada, Singapore. And you see the distribution, academia, industry, networks, non-for-profit organization, particular patient organization. And the good thing is, it's not just a passive LOS, it's really active contribution of the whole Restore community to, for example, to the task of the 17 working groups in refining the roadmap. More than 250 were involved, and to each working group is a range between 26 to more than 100 contributors. And that, I think, is a good success. That really is a contribution by different peoples and different institutions. Our vision is clear. We want to make the disruptive promise of advanced therapies a reality to cure chronic diseases and make advanced therapies accessible as a standard of care for every patient in need, and make use of the enormous socio-economic potential of novel breakthrough therapies. And to get this, we have to overcome the technology and regulatory roadblocks for advanced therapies in Europe. We want to deliver a pipeline of dozens of revolutionary IT products by the next 10 years and establish a sustainable pan-European ecosystem integrating research, clinics, industry, and of course, very important, the patient view. What is Restore aiming for? I think we want, of course, to accelerate and de-risk the clinical development of advanced therapies by overcoming the roadblocks, by joint efforts, to have or to get more ATMPs in the future, faster, more cost-effective, and less failure. The question is how to get there. And we think there are a couple of pillars what we need. First, I think we need a coordination and support activity, which really helps the Board of Funders, the European Commission, and the member state authorities really to figure out the roadmap. That means a board consisting of representatives of academia, industry, biotech, pharma, but also technology providers is very important, and patient advocates. Interaction with the payers at national and European levels, but also with the regulatory authorities, regulatory bodies, both at national and European level. And the aim is clear, a consortium with a transparent and dynamic governance that provides support, guidance, education, advice, networking, and expertise for the large-scale research initiative, representing the academic, industry, and patient society. And we think there are different elements: structured research and innovation actions, infrastructure, technology research and innovation platforms, and private-public partnerships, including SME. I will come to this. I think one important point is, if you compare, for example, with China or with US, there's a forming of very big centers, and we think we can take advantage of both things. First, there is a clear advantage of scattered European research ecosystem, because there's a clear data analysis from the group of Wu, recently published in Nature, showing for the last 20 years what was the impact of different kind of research on the society. And it's absolutely clear, think small if you want to have creativity. Small teams are much more creative, take more risk, and therefore most of the creativity comes from small teams. But on the other hand, if you really want to bring innovation, to bring the things to the market, you have to think big. And I think what we need is really a combination of both. That means to combine the benefits of small science teams scattered across Europe with their breakthrough discoveries, and centralize large transdisciplinary teams to translate innovation. And we think here the structure of incubators or hubs are very important, close to patient care, transdisciplinary also with ethics, patient advocates, pharma and tech industry, IT, data management, health economy, and so on. And that is a docking point for the pharma and tech industry. but also for selected projects with high translational potency coming from inside or coming from all the different small groups. And that is, I think, a very important point. And how you getting the ideas, and one element is, of course, the research and innovation actions, the typical RIAs in the European program. Structured RIAs, we think, according to the roadmap, feeds the small teams to generate mission-driven, disrupting science and technology, interlinked in thematically structured small consortia, keeps the RIA project flexible that can be halted or extended based on key performance indicators. And that, I think, is a big difference to the current strategy, where you have fixed 4 or 5 years and nobody take care what is the result, positive or negative, and you have no chance to continue. And the final aim is to really have a European research area on advanced therapy, to building up a project pipeline, delivering disruptive solutions. And of course, I have no time to go through details. We have time here in the 2 days to discuss different points. And just as an example, pipeline projects, I think very important is to uncover and tackle more rare diseases, particularly also beyond hematopoietic stem cell repair, because now we have access to different adult stem cells in the muscle and the liver and so on. And but also the iPSC technology to repair genetic diseases, completely different sources. From rare disease to common diseases, particularly going to solid tumors, to immune diseases like autoimmune diseases and so on, which is a huge indication, cardiovascular diseases and so on. From treatment of chronic diseases to preemptive approaches, I think is a very interesting aspect that you can treat patients at risk, for example, after trauma, ischemia, reperfusion, or with a particular risk genetic background or therapy-resistant acute infections before it go to chronic one. And we need also exemplary new technology projects, in vivo gene therapy, steerable, tissue-specific gene expression, targeted gene editing, and the problem to solve the problem of immunogenicity of, for example, adeno-associated vectors or other viral vectors. The problem with CRISPR-Cas that we have a high immunogenicity of Cas, and also sometimes of the transgenes in case of rare diseases. Ex vivo cell and gene therapy, universal cells, including allogenic off the shelf, but also cells which have a better fitness. Preclinical models, I think there's a complete change in the paradigm from mouse models to, or primate and so on, to human on patient on the chip technologies, and it's a very important aspects for the future. And finally, of course, manufacturing, automation, control by artificial intelligence, also regarding to the response in the patient, and also composite products, meaning particular with biomaterials and matrix structures. Also, therefore, we think we need to building up research and technology innovation platforms and cross-team platforms to prevent that everybody reinvents the wheel. And we think, and you have seen it in the working groups, we discussed about the pipeline project platform, preclinical technology platform, manufacturing technology platform, and so on. And also cross-team platforms for ethics, for health economics, and product and therapy optimization, which is a major aspect for big data management and artificial intelligence. And at the academic hubs, the element of refined translation to de-risk the problem after a late stage clinical trials by very in-depth characterization of patient response and product in the early phase. And I think last, new models for PPP and SME support, I think strengthens the relationship of academia to the industry, pharma and biotech, but also very important, the technology providers for solutions in cell and gene therapy handling, together with the academic incubators and hospitals. And don't forget that we focus on patient-centered care, that meaning coming from the medical need, and also support of SMEs. We discussed with the European Commission so-called special interest venture funds targeted to such kind of direction. They think that is an opportunity to make a co-financing for private money in this particular fields. What is the impact? We think RESTOR can have a huge impact, or in particular this technology of advanced therapies can have a major impact. Of course, on science and technology, we can build up this infrastructure of translational centers of excellence for the future, which is very helpful for the patient, but also for the industry for docking stations. Boosting Europe's competitiveness in the groundbreaking area of living drugs fundamental knowledge and technology breakthroughs relevant for many areas, meaning we can use the tools also for other things. For example, the in vitro models, human on the chip, of course, is also useful for conventional pharma development, but also in other fields. Next generation of translational scientists, which new mindset, really looking from patient to bench to bed, and bed forward and backward. But we have also a lot of benefits for society and economy. Improved survival and quality of life for patients with chronic diseases is, of course, a major aspect and motivation. And by this, decreasing the financial burden of society of chronic diseases if we can cure patients. But also to generate an innovative product pipeline for medtech, biotech, pharma industry, and completely new players in this field. And you see the examples, for example, California Institute of Regenerative Medicine, that was a vote by the taxpayer 10 years ago to invest, in summary, $3 billion over 10 years. But the good thing is, after 10 years, they have a reimbursement or whatever revenue of 6 times more in investment in California industry, showing that before any product was approved, because it started very early with stem cell approaches, and they have more than 60 trials running, but most importantly, they have a lot of working places installed. A similar model in Canada, the CIRM, showed now even a ratio of 1 to 10 between investment and return of investment before even these first 2 points come a realistic point. Therefore, I think it's very worth to invest in this field. All generation of highly specialized workforces and expertise to keep it in Europe and not that they leave it. And setting ethical and quality standards, that is very important because there exist very obscure institutions. In summary, what we recommended to the European Commission is switch from the current focus on scattered, time-limited and unconnected EU funding to a concerted funding program for advanced therapy with innovative elements supporting a creative and productive ecosystem. And we try to motivate them to take the momentum gained from the FED flagship initiative. You know, we started with this, and bringing together the stakeholders in the field and go new ways by supporting sustainable structural elements with sufficient budget according to the achievement of key performance indicators over the next 7 years of the Horizon Europe program, which starts in 2021, as an adequate structure and financial alternative to the cancelled FED flagship program. You know about this. And as a point I already figured out before, the pillars of this kind of recommendation. Okay, what can you do? What can we do to make Restore flying? I think on one hand, the refinement of the roadmap, which is almost done by the consortium, and we will publish it very soon and deliver it to you for a final approval. And of course, I think we need really activity in the science policy and political support, both by stakeholders at national and regional level, which then directed to Brussels, and by stakeholders at European Parliament and European Commission in Brussels, with the aim clearly that this topic, advanced therapy, have a key role in the new Horizon Europe program. You know, it will be finalized, at least the frame, within the next couple of months, because probably the new commission is working December 1st. Thank you very much for your attention. And again, enjoy the meeting. And it's now my pleasure to introduce the next speaker. Yes, was Ami. Yeah, Ami Appelbaum, and Peter already introduced him. Thank you very much for coming and giving a talk on the Israel view on supporting advanced therapies. Thank you, Dr. Appelbaum, for coming. Let's move forward. No?
Yeah.
And if you need a pointer, the green one is better.
Okay.
The first one, no? The green one. Thanks.
Good morning, everyone. Oh, you're still asleep, huh? Good morning. I hope my presentation will come up, which here it is. Maybe let me introduce my position, and so you know who is talking with you. So I'm the chief scientist in the Ministry of Economy and Industry in Israel, and also chairman of Israel Innovation Authority. And you may ask why Israel is represented here. So the previous speakers already introduced Europe and mentioned Israel. And now I would like to explain to you what the impact that Israel has or potentially can have on this consortia and its activity. Israel Innovation Authority is a body that is bridging between the public sector and the private sector. In simple words, we take public money, we provide it for research and development for the private sector. What is important to know that we are going through a major inflection point. Others call it disruptive, but I don't want to. Disruptive is kind of negative connotation. I would like to talk about positive. It's inflection point because every segment of our life is changing. If you look on the artificial intelligence today and the computer and robotics, you see how much they're changing the way we think, the way we behave, the way we get medical services, the way we get the industries running. Our life is changing forever. And I would argue it's one of the largest changes that we experience throughout humanity since the first person on Earth. Look how the agriculture is changing. And you know that if by 2050, if we will not double the productivity of every acres in the agriculture, we will not have enough food on Earth. So we are changing quickly with the precision agriculture to double the productivity. If you look about the financial world, is changing. If you look about driving from point A to point B, if you don't believe me, go ask Daimler-Benz, BMW, Volkswagen, what they think about their industry. They will tell you, if we will not change from where we are now in 5 or 10 years toward the electric autonomous car, we will lose our industry. If you look about real estate, if you look about how we even plan our trip, everything in our life is changing, and it's all depend on innovation. Those who will not innovate will be under the wave and will be dumped. Those who innovate and take their innovation into the market, those are the ones who prevail. The whole history may change, and those who were strong 5, 10, 15 years ago may be very, very weak 5 years, 10 years from now. And that's what we want. And Israel, as you know, although a small country, but as you can see, big passion and great success that I would like to share with you. This change in the market, where the world changed from the physical product, you can look on the left side, 2007, out of the top largest companies in the world, 5 of them were in oil and gas, 5 of them. Fast forward to 2017, none of them is the largest company in the world anymore. None of them, not BP, not Exxon, they disappeared from the top 10. The largest company now are those who are with data-driven, with virtual product, as we call it. Now, where Israel getting into this? Israel, in 2007, had only 4 of the largest company in the world has a research and development in Israel. The small Star of David is indication where Israel, those companies has R&D in Israel. If you look 10 years forward, all 10 top companies, largest company in the world, have research and development in Israel. And you may ask why, and that's why I'm here, to try to explain to you. So first, what you want is a strong academia. And you can see that Israel is #1 in quality of scientific research institutions. #2 in technology incubator startup. #5, so strong academia, basic research is a prerequisite for any innovation and any success. Second, what you need is capital. And you can see investment in R&D as as a percent of the GDP, Israel is number one in the world. Actually, today, 2019, Israel is second, Korea is number one. 4.3% of GDP in Israel goes into R&D. And if you look on how many VCs, meaning foreign VCs mainly, you can see again, Israel in a percent of GDP is above even the US. In the last 2 years, that's yield $11 billion investment in Israel of VC, and more than $40 billion of exit by those companies. The third element that you want is to have a multinational companies. Again, Germany is a huge country. Israel, very small and surrounded by friendly neighbors. So we can always go to our neighbors, to do what? Not to do research and development. So we need those multinational companies to come for two reasons. One, because they're bringing a lot of technology and creating a lot of jobs. Second, it's helped Israeli company to go out. And you can see what I said before about change in the world, the way the economy is operating in the world today, that if in the 60s we had few companies in Israel, multinational, as of 2000, when the switch started to go into the digital and data science, then exponential growth in the number of companies that coming to Israel. And I can talk more on and on that. You can see automotive companies here, and you may ask why automotive companies like Ford are in Israel, General Motors. The reason is that Israel today have more than 500 automotive companies. And you ask, why? What? We never heard about any car that made in Israel. It's good you didn't hear because we had once and it shut down. But we have many 500 companies who deal today with the future autonomous car, cybersecurity for automotive, controller for electric cars. Those are all being driven by the change in the automotive industry, and therefore Volkswagen, Daimler-Benz, BMW, General Motors, they all have research and development in Israel, and what we want, that they will develop those research and development more into more engineering. We don't want to assemble cars in Israel. I tell you why. Because the assembly actually is not where the highest value is. The highest value today, it's in the content of what you do. And Israel changed dramatically in the past 34 years. As I will go quickly, the population doubled. The GDP per capita was 5 times, almost 6 times higher. So standard of living went way up. You can see number of cars per 1,000 inhabitants. It went from 15 to 365. That's more than 20 times. Even the defense budget as percent of GDP went down. And you may say, of course, because your neighborhood become more friendly. No, don't misled by that. It's not become more friendlier. It didn't become friendlier. It's because the GDP went so much up. In absolute terms, the defense went up, but as percent of GDP went down. And you can see the export, overall export, is $100 billion. And now, actually, this year will be 114 billion. I just read it yesterday. And the high-tech is leading. It's say 23. That was in 2018. Actually, it's closer now to 30 or 40 billion dollars, just the high-tech industry. So the Israel become a global innovation and entrepreneurship. If you have the academia, if you have the capital, if you have the government that understanding, and you need the culture of out-of-the-box thinking. Dare to think big. Don't be afraid and move fast. That's part of our culture. Government is supporting all of that. And that's the Israel Innovation Authority. As I said, our job is to promote and sustain innovation and move to the inclusive growth. We are strengthening innovation ecosystem, we are enhancing the economic impact, and we are enabling emerging disruptive technology. That's the overall picture about Israel as a whole. The way that Israel Innovation Authority does it is, first, if you look at the bottom here, we are funding every field and every topic that innovators or entrepreneur or company want to invest in. And you can see that we're supporting cyber, robotics, aeronautic, education tech, transportation, pharma. We fund it if there is somebody who want. The way that we fund is a matching fund between the government and the entrepreneur. I'm short on time, I will not go into all the technical detail. And we fund technology transfer from academia to industry. We fund entrepreneurs, we fund startups, we fund SME, as long as they come with differentiated technology and as long as they have a global strategy from the get-go. Because Israel is a small country, we stay neutral, and we don't take equity in the companies. And if the companies succeed, they give us back the loan in a very, very slow pace, 3 to 5% of the annually of the revenue. If they fail, the money, the loan is being forgiven. If you move on, so what is the largest, fastest growth element in Israel today is artificial intelligence. And that artificial intelligence is a platform for pharma. You can see here the investment from 2015 to 2018 in AI grew up by almost 60-70%, and number of companies in those 3 years almost doubled, increased by 100%. You can see that that's artificial intelligence also lead into a lot of growth in the pharma, agrotech, digital health. Those are the area. Of course, we have cyber, but digital health, pharma, agrotech, those are the area that we all talking now in life science. 30% of Israel Innovation Authority fund, as you're going to see, is going into life science. So the whole medical world now, the whole life science is changing dramatically. And this change create a very significant ethical situation, regulation, that what we think we need to address from day one. Bringing the regulator, bring in the people who define policy, many ethical issues that we need to think and rethink early on from now, and not to let it drag and come out 5 years after the product ready and everyone complained that our privacy was compromised and we give big fine to companies. If we bring in those regulators and those policymakers and those philosophers who look ethically what's happened, that will be absolutely the necessity of the way to proceed. If we move on, the whole ecosystem you need the government to understand, and Israeli government understand the need for research and development and for growth. You need the academia and very strong, you need the industry, and you need the healthcare system. If you don't have it all, you cannot make progress. The way that the medical industry in Israel is distributed in the manufacturing, you can see medical equipment is 43%, about 50%, and then digital health and biotech is another 30% more or less each. If you look on the growth that happened in Israel, you can see here in the life science, you can see the investment that go for the seed company, for the R&D, $578 million, and for the initial revenue, and those companies with revenue growth. So you can see from the 2008 in 10 years, it's more than 4 times growth in Israel life science industry investment and growth. Israeli government passed a resolution to invest $275 million for 5 years to make the national growth, to become what we try to be, a world leader in digital health solution. We want a clinical and academic research, and we want that there will be the growth engine. And you say, wow, with 275, you think you can change the world? The answer is those 275 million are just the seed, and companies and organizations can leverage it, and by our experience, 10 to 20 times. We see when the government put the money where there is market failure, the industry, the private sector, taking over and can multiply it by 10 and 20 times, and we see it. You can see in the digital health that we added like 500 company in 6 years, in 7 years. You can see here the growth, the fast growth of digital health company. You can see the investment in the digital health. So digital health, it's definitely one of the vehicle growth engine for Israel industry. You can see how many different company in medical imaging, in genomics, security, analytic, and so on. It's just illustration that it's loaded with activity and many, many companies. One of the area that we would like to do as a country is the national database, which we called it Mosaic, for genomic, clinical, and biopsy. The healthcare provider will collect from volunteer samples. They will sign on agreement with the volunteers, and we will collect all the data, genomic, clinical, and biopsies, that we already have data distributed among the health provider going back more than 20 or even 30 years. For I think 85% of the population in Israel, we have the data that looking 20 years back. Think now that you can look forward and do analysis on all that data, both people who are still healthy, those who are sick, and get a lot of analysis. In the pharma, the Israel academia actually generated a patent and molecules that eventually yielded $29 billion of revenue. Unfortunately, most of it was created, the money, the IP was created in Israel, but eventually companies who took it out are non-Israeli. And you can see that out of the $29 billion, you can see here the company, only $6 billion were developed in Israel. So show you what happened if your industry is not leveraging all the potential that the academia has. It is complicated to move knowledge from academia to the industry, and that's what we're all trying to do. But look what impressive number of almost $30 billion that is now being sold on patents that was created here in Israel. So the last point I want to talk, which you already started to hear, is a combination between the biologist and the data scientist. And if you look on the healthcare provider, the biopharma, the medtech, and the therapeutic and devices, if you take now the digital health and you integrate all of that, you go into a bioconvergence. Bioconvergence is to help growth of different industry, leveraging the biology, leveraging the data, and the artificial intelligence, and look how the medtech you can develop it, digital health, biopharma, and moving in life science, which is still not in the health provider, but agriculture, energy, and food tech. And that's all of those will be in, you can make 3D organs, immune materials, and so on. So in summary, we have the information is today a strong power. We are going into startup and growth company, and we're developing 5 years programs. Thank you very much.
Thank you very much for this.
Thank you.
Very impressive overview. I think it's really a role model how it can work, and I think Europe can learn from this model. Okay, I think we move forward in the scientific program now, and I invite the chairman of the first session.
So also a warm welcome from our side. Annelies Bienencourt and myself, Georg Duda, are happy to chair this session. And first I'd like to congratulate Petra and Dieter for organizing this event and bringing all the people together. And we're looking forward to the various presentations. After that inspiring introduction of the Israeli capabilities, we're going now into the more basic research possibilities towards new therapeutic and diagnostic possibilities. And the first talk will be by Alexandra Marquet. And she will talk as a research scientist from Minho about on the way to improved therapies for skin diseases and disorders. And thank you very much for being here.
Thank you very much for the introduction. Good morning, everyone. First of all, I would like to acknowledge the organizers for the opportunity to show here the work that we have been developing in the Trebis Research Group in Portugal at the University of Minho, and to highlight a little bit the importance of skin tissue and wound healing for this in the context that we are talking about. So it is, in fact, this is not working? The green one is for going on forward. It's not working? Okay. Okay, in fact, despite the hundreds of dressings and some cellular products that are available to treat skin wounds, we still have these situations, these chronic wounds. Sorry, it's not working very well. Anyway, we still have chronic wounds that either do not heal or take years to heal, and that clinicians are not able to deal with. And if we associate these chronic wounds to diabetes, and this is just a part of the chronic wounds, we can see that 15 to 25% of the diabetic patients develop this type of ulcers. And if you look at the numbers that I have here, in 2017, we had about 400 million patients with diabetes in the world, and the expectation is that by 2045, this number rises to more than 600 million. So it's clear that this is a problem, and as I said, it's not the only type of chronic wounds that we know of. In addition to chronic wounds, there's another critical issue that are the wounds that result from burns, and although the numbers are not as high, let's say, as the chronic wounds, the mortality that is associated. Great, thank you very much. Yeah, okay. So the mortality associated to this patient is really striking, and this is because when people is burned, the barrier is disrupted, and the probability to infection and to dehydration is really high, so leads to very high mortality rates. So in our group, we have been looking at this problematic, mainly focusing on two approaches. One that we called natural extracellular matrix approach, and the artificial one. This one, the natural one, is based on this technology, cell sheet engineering, that was first developed in Japan by Okano's group, and relies on the formation of sheets of cells that can be of different types that we can then pile up and build 3D structures. So this is a fully biological approach, which allow us to address different questions that without the interference of the biomaterials. On the other side, we have this artificial approach that is based on a processing that we patent. We can obtain these porous structures that can hydrate very easily and can be very useful to manage the wound exudate, for example, and that retain properties that are quite interesting in terms in relation to the hydrogels from where they are produced, but are much more mechanically stable, and importantly, they provide a different support for cells. So just starting with, I'm going to give you one example of each one of the approaches that we have been exploring. And when we think on diabetic wounds, we know that the pathophysiology is very specific. There are very particular aspects of this wound, like impaired angiogenesis, really imbalanced matrix degradation and deposition. So it's really critical to address these specificities. So what we approached these wounds was to use these structures, the spongy-like hydrogels, that we combined our polysaccharide, the base polysaccharide with hyaluronic acid, that we saw in a different study that when degrades leads to the formation of low molecular weight fragments that are angiogenic, and we combined this with adipose stem cells. But we did another approach. We preconditioned these adipose stem cells in order to lead them to secrete some neurotrophic factors, aiming to focus on the lack of innervation of these diabetic wounds. So when we looked at, so we had these three different conditions, just the material with the adipose stem cells and the conditioned ones. And when we looked at the wound closure, we didn't see significant differences. As we saw in other studies, it is interesting to understand that when we have a material in the wound that we do not intend to remove, that we aim to let it there until the wound to close, we really see some delay in the healing, because the material is actually somehow impairing the reepithelialization at early time points. So when we looked at the neovascularization, as we hypothesized, when we had the hyaluronic acid, we actually saw a significant difference in relation to the control. Although when we have the adipose stem cells, we could not see that difference. Again, potentially because the ASCs and the matrix that they are depositing is somehow affecting the degradation of the material and somehow impairing this result. But when we looked at the inflammation, we were actually quite surprised when we looked at the anti-inflammatory and the pro-inflammatory macrophages. We could see a different profile when in comparison with the control, our experimental conditions. And the most interesting result that when we looked at the ratio between the anti and pro-inflammatory macrophages, we could actually see that there's a significant difference in this condition where the adipose stem cells were preconditioned to the production of the neurotrophic factors. And this means that with this ratio, the transition to the proliferative stage of the healing was faster in this condition in relation to the others. And then when we looked at the neo-innervation, that was one of our hypotheses with the preconditioning of the ASCs, we could not confirm that conditioning to the production of the neurotrophic factor had a significant effect in the intraepidermal nerve fibers that were formed. In fact, what we saw was that there is although not significant, a number of the fibers that were observed in this condition that was not the preconditioned one. So what we concluded from this study, and that's something that we are looking at in more detail, is that we really need to have a well-controlled transition from the inflammatory to the proliferative stage of the healing in order to be able to have all that to restore the full functionality of the tissue. So now moving on into the strategy that we use, the cell sheets and the cell sheet engineering. In this case, it was a work that we performed some time ago. We combined different sheets of adipose stem cells, and here we compared basically these thermoresponsive dishes that are the ones that are originally the basis of this technology, and sheets of cells that were just peeled off from the bottom of the wells. And what we could see is that first, immediately, the constructs that we obtained were much more stable when we obtained the cell sheets from the thermoresponsive dishes. And when we looked at the tissue the neo tissue that was formed after the transplantation in full thickness mice wounds, we could see that we had some effect only in this condition, the one with the thermoresponsive dishes, in the neoepidermis regeneration or formation. So we went a little bit deeper in trying to understand what was going on, and what we could see was that, in fact, in this condition, with the thermoresponsive ones, we had a higher expression of keratinocyte growth factor. So at that time, we proposed a mechanism in which the ASCs were actually directly interacting with the keratinocytes at the wound margin, and they were sustaining this loop of KGF-driven response, and then because of that, we had this impact in the neoepidermis that was formed. Currently, we are trying to understand if this is also because those studies were done in mice, and at the moment we were doing the same experiments but in pigs. And we confirmed that in relation to the control, when we had the cell sheets, we actually had an impact in the reepithelialization. We had a faster reepithelialization with these structures. But interestingly, and it is something that we are still looking at, when we used human here and porcine cell sheets, the results are completely different. We only got a significant difference in relation to the control when the human cells were used. So at the same time, we are trying to understand exactly what is the mechanism in confirming the involvement of the KGF and this specific interaction between ASCs and keratinocytes. And here you can clearly see that when we have adipose stem cells in contact with the keratinocytes, we have a significantly faster closure of the gap that we made in this layer of keratinocytes. And here, when we use the conditioned medium, we can see clearly that the migration of the keratinocytes, it's completely different in relation to this condition where we use the antibody against KGF. So basically, we are demonstrating that keratinocyte growth factor is involved in this response. And we can see here even further and looking at and going into what we hypothesize, that there's really an interaction between the two cell types. When we have the cell sheet they are really sticky and they can adhere to the wound. And here we can see that when we have suspension of cells or a cell sheet, we have different results, and with the cell sheet, this response is even higher. And we just confirmed this in ex vivo 3D experiments, and you can see here that when we layered the cell sheet of ASCs on top of the wound, this is the wound, we can see the epidermis being regenerated, and again, the amount of KGF significantly increased in relation to the control. So this is very interesting and shows different approaches with the adipose stem cells, and what we have in the clinics and that the clinicians have available regarding cellular products are not similar to what is actually the skin. Skin is a complex organ formed not only by epidermis and dermis, that are what is available out there, but we have the vascularization, the immune system, the nervous system, we have all the appendages, and we also have the adipose tissue. So what we think, and in addition to the stem cells and the importance of stem cells in wound healing, is that maybe we really need to go a little bit more into more complex structures in order to have an alternative to the current cellular products that are transplanted and integrated into the wound to form a new tissue that is fully functional. So The better approach, or the best approach to look at this complexity, of course, it's 3D printing. I must say that I think that we are still very far away, but anyway, this is probably the best technology that we have to address this complexity and to try to look at the different compartments that skin has. And we have been also looking at this from different approaches, and we have been developing our specific bioinks that we aim to target for the different types of cells that we have in skin. And for that, we have been coupling different peptides that are present in the different proteins of the ECM of the different compartments. And at the moment we have epidermis, the vascularization, the dermis, and we are looking also to the adipose tissue. At the moment we are combining all of this in order to have the more complex structure. But we are also looking at the compartments, and I'm not sure if this will be used in the complex structure, but I think tissue engineering is also very important, and these approaches are also very important to generate the in vitro models where we can address specific scientific questions and that allow us to understand what are the conditions, how far can we go in mimicking this complexity. And using the same rationale in the hair follicle, we are looking at the dermapapilla, and here we have two main components, the mesenchymal and the epithelial compartment. And I'm just going to give you an overview of what we are looking at, and the same approach, we are following the same approach, looking at the extracellular matrix, here for example the collagen 4 and the fibronectin here, and we are coupling or grafting the different peptides in order to see what is the effect in the dermapapilla cells in this specifically mesenchymal compartment. And the issue with the dermapapilla cells is that when they are removed from the native tissue, and if they are cultured in 2D, they just lose their inductive properties and they lose their capacity to form hair. So the challenge first is to have a 3D environment in which these properties are restored, and they can then, after transplantation, to give rise back to the hair follicle. So we have been looking at how they deposit the matrix, and what I would like to call your attention is to the ALP staining, the alkaline phosphatase, that is one of the markers of the dermapapilla functionality, and we can clearly see in relation to the control that here is just a qualitative analysis, but we have a partial restore of this functionality. And if we look at the quantitative data, we can, of course, we have been looking at different concentrations of polymers, different conditions, and you can clearly see, for example, here are the two peptides, the RGD and the EP3. We can clearly see that we have a different profile when we have the two peptides. So definitely we are somehow manipulating the phenotype of the dermapapilla cells. And just to finish, we have also been looking at the epithelial compartment, and in this case, some years ago, we proposed a population of epithelial cells, let me call it like this, that they are not epidermal stem cells. They have some characteristics of early differentiated keratinocytes that we isolate from a full population of primary keratinocytes from a skin biopsy. So they are very early in terms of phenotype, in terms of maturation. And at the moment, what we are trying to is to propose this population in comparison with the hair follicle keratinocytes that are not really available there to generate these structures, or to generate hair follicle models, to try to propose these cells as an alternative. So we have looked at the phenotype, they are quite similar, and importantly, when we transplanted these cells together with dermapapilla cells, we could see as we saw with the hair follicle keratinocytes, the formation of these epithelial related structures that seem to be promising in terms of having this subpopulation as an alternative to these keratinocytes that I've been using so far. So just to finalize, to acknowledge our funding, the international collaborators, of course, many of them very critical for the foundation of some of these works, our national collaborations, especially our clinicians, and of course the team of researchers that have been involved. And thank you for your attention.
Thank you very much. Are there questions? Yes, please.
So thank you very much. Very nice talk. So concerning the ASMs for the diabetic wound healing, if I get it right, the active component is the KGF, which is secreted by the ASMs. And I guess you go for an autologous approach. So if you would now test different ASMs from different donors, would you then see that they are secreting the same amount of KGF? So is it standardizable? And if not, would there be another way to secrete or to provide the KGF to the wound?
Well, that's a very good question, because one of the issues, even for diabetic or even for burn patients, is that the profile and the ASCs are not exactly the same as these ones. These ones are from healthy patients, and those are the samples that we have available. And I'm sure it will be different. What we are doing now is to actually develop a biomaterial-based strategy to deliver the KGF and to see if that is really getting to the same results that we are getting with this. Yes, thank you.
Probably just adding another question along the same line. You indicated different sort of skin wound initial situations, which I would expect have different inflammatory environments after a burn or after a diabetic. Do you think that one cell type could address all those different inflammatory settings, or how is the vision there?
No, I'm not a believer of one therapy will fit all the types of wound. I definitely think that we really need to address the specificities of each wound. And one of the issues with the approaches that we are trying to develop, and we worldwide, is that we do not really have, for example, animal models for chronic wounds. So this is a big drawback. So it's very difficult to look at that in particular. But yes, definitely we need to address specifically each one.
Thank you very much. And I think we need to move forward. Thank you.
Okay, thank you.
Well, thank you. So we will continue. The next session, talk will be Andrea Kögg from United Kingdom, from Catapult, and his talk will be targeting the tumor vasculature with CAR T cells for treatment of solid tumors. So thank you. Andrea. Okay. Okay. Thank you. You have 10 minutes for talk. Thanks.
Okay, so hello everyone. Today I'm going to give a brief overview on a project that we work with within the Cell and Gene Therapy Catapults. So the title of my talk is Targeting Tumor Vasculature with CAR T Cells for the Treatment of Solid Tumors. Okay, so firstly, I just want to give a bit of a background on the Cell and Gene Therapy Catapults. So we're part of a world-leading network of technology and innovation centres that have been set up within the UK to drive innovation and economic growth. We have unique technical facilities and expertise to help adopt, develop and exploit these innovations. And we bridge the gap between businesses and academic research. And we were initially established in 2012 by Innovate UK as a not-for-profit independent centre. So our mission or aim as such is to make the UK a global leader in the development, delivery and commercialisation of cell and gene therapy products. So firstly, just give a bit of a background into chimeric antigen receptor CAR T-cell therapies. So there is 2 FDA-approved autologous products on the market at the moment. So you have Kymriah that targets AAL, and this is from Novartis, and then Yescarta that looks at non-Hodgkin's lymphoma, which is from Kite or Gilead. And although these have been quite successful, there is still a lot to do with solid tumours. So this talk will primarily look at targeting solid tumours with CAR T-cell therapies. So there's many challenges of targeting solid tumours, so I'm going to name a few here. So the first one is the access to the tumour tissue, the lack of appropriate target antigens, and also the immunosuppressive tumour microenvironment. So what we aim to look at is how to approach this. So one therapeutic or potential therapeutic approach is to target the tumour angiogenesis. And with this, we want to look at a specific tumour endothelial marker called CLEC14A. So this is the C-type-lectin family. It's mainly expressed in tumour tissue compared to healthy tissue. And you can see with 3 different carcinomas here on the left in the staining. We have breast, liver, and bladder, and you can see that CLEC14A is stained in green, and it's highly expressed in each of these models, but when the corresponding healthy breast, liver, and bladder tissue, there is no expression of this marker. We have been working in collaboration with the University of Birmingham in the UK, and they have done a lot of the preclinical animal models to look at this target. So they've generated CAR T cells to target CLEC14A using gamma retrovirus. And you can see here that in a RipTag2 mouse model, we can see that with the CAR T cells, there is like significant reduction in CLEC14 expression when you use the CAR T cells compared to the mock. When we quantify this as well, we can see that the percentage S cell area is quite reduced when we use the CAR T cells. The percent of vasculature expressing CLEC14A is also reduced compared to the mock treated animals. And then again, when we look at the percentage of vascular expressing cleaved caspase 3, we can see a high amount of this within the CAR treated animals compared to the mock. We are also quite looking into the toxicity of this treatment in the animal model. So we went to look at 2 different CAR treatments against mock, and we could see that days post T-cell infusion, the weight of the animals was not affected. So we were quite happy with this. So to summarize the background into this project, we are looking to target tumor stroma represents an attractive approach for the treatment of solid tumors. We have seen 50% reduction in tumor burden achieved using CAR T cells targeted CLEC14, and we have tested this in 3 different models. And also there's quite happily no signs of toxicity in the mouse model. So within the cell and gene therapy catapult, we took this preclinical data and we wanted to look into having a more safer, effective approach. So we looked into not using the gamma retro CAR T cells and more CAR mRNA, so transfected. So we could use this in a first-in-man trial. So to do this, we were generating large-scale generation of mRNA CAR T cells. And to do this, we used Prodigy from Miltenyi. So this is an open system, and we can do all of the processing within this, and a design of experiment approach was used for this processing to look at what pulses, voltages, time, the concentration of the CAR mRNA, and the concentration of the cells that we would use. So just to go through it in brief, we use our starting leukapheresis material. We would then do a CD4/8 isolation, culture the cells in the presence of activation or non-activation. We would then electroporate within the system and finally harvest. And from some of the runs that we generated within the cell and gene therapy catapult, we can see that with both activated and non-activated T cells, we are getting quite high cell viability, which are in the grey bars, and transfection efficiency also. And there is no real difference between the non-activated and activated. There is slightly higher viability and transfection efficiency with the activated T cells, but if we were to use this in the clinic, if we use non-activated, it would greatly reduce the time to clinic. So do these CAR-mRNA T cells work? So to characterize the process, we in the analytical development team in cell and gene therapy Catapult have looked at 3 main outputs. So the first one is a prime flow RNA assay. So it's a flow cytometry approach where we can look at CAR protein and mRNA detection in the same sample. We also use a droplet digital PCR assay, which we can look at for CAR-mRNA detection and persistence. And then finally, as a QC release assay, we looked at impedance-based potency assay. So this is a label-free real-time killing assay. But for the purpose of this talk, I'm just going to focus on the ddPCR assay and how we developed it. So again, like with the processing, we use a design of experiment approach where we can look at numerous factors. So what we tested was, so we designed 4 primer probe sets against the CAR. And within that, we looked at primer concentration, probe concentration, and annealing temperature. So we looked at a low, a high, and a center point concentration for each of the factors. And the RNA template that we used was a T cell background spiked with the specific copies of the CAR-mRNA. And as controls, we had the CAR-mRNA alone and T cells which shouldn't express the CLEC14A. So the main results comes out as a response surface model, and this is just one representative image of what we typically get from this design. We also, in this one here, we looked at primer-probe interaction, but we also have results for primer annealing temperature interaction and probe annealing temperature interaction. And as you can see, what we wanted to get from this is to have quite a large separation between positive and negative droplets within the droplet digital PCR assay. So you can see this in the delta fluorescence. But as the probe, the primer doesn't seem to have an effect within this model. When you increase the concentration, it's mainly the probe that gives you that greater separation. So we had a number of different combinations to test this and confirm what we'd seen in the DOE. So we then decided to test a number of these, and this is just a graph that represents what we've seen. So the expected copies as 14,000 copies are defined by the red line at the top of the graph. And when we look at T cells and with the CAR-mRNA spiked and CAR-mRNA alone, we get roughly the same recovery of copies of around 57%. While doing this, we were thought this was strange that we weren't getting complete recovery of the copies that we spiked in. So in parallel and not with the actual confirmation study, we went to look at using a 1-step versus 2-step approach. So we went to look at anti-CLEC14A mRNA copies with 1-step in blue and 2-step in pink. And although you can see with the T cells and CAR-mRNA, we had higher recovery with the 1-step. With the CAR-mRNA alone, it was the complete opposite. But for consistency purpose, we decided to go with the 1-step approach. We also then went to look at linearity. So we looked at with the CAR-mRNA alone and spiked into a T cell background, but we also had the opportunity to look at transduced versus non-transduced. So we looked at different ratios of transduced to non-transduced T cells, and we had quite good linearity. We then looked at multiplexing with a reference gene. So we tested a number of reference genes to see which was more stable in activated and non-activated T cells. And finally, just to quickly end on this, we've seen that in 3 donors, we had quite good expression of this reference gene. And when we multiplexed it in this assay, we got quite good multiplexing, and the single-plex and multiplex was quite comparable. So in summary, we've developed a ddPCR assay capable of detecting CAR-mRNA, and we're currently qualifying this assay to demonstrate the specificity and robustness of this assay to be used in the clinic. So this is just to give an acknowledgement of all the work that's been done with the University of Birmingham, with Cancer Research UK and also with the Cell and Gene Therapy Cash Pool. So thanks for your attention. If you have any questions, I'll be happy to answer.
Thank you very much. So there is some time for one question. So. Okay. One is here.
Yeah, good morning. So may I ask how close you are to the clinical trials with this approach?
So we're currently doing a lot of the process development work at the moment to generate the amount of CAR T cells that we want. So hopefully soon we'll be going towards MHRA with this.
There's one question in the back.
So Beatrice Mintz has shown quite some time ago that you target exclusively the tumor, the stroma of a tumor, then the tumor cells become more aggressive. Do you see that also, or is it a danger?
So we weren't particularly involved in the preclinical animal models, but it's just that we decided this one would be a good therapeutic approach because we can choose we can target the antigenic potential of the tumor. So it's mainly that was the target of this therapy.
Okay. Thank you very much. So we have a short cafe break and we can back to 10:50. Thanks a lot.
Welcome back, everybody. If you could take a seat that we can restart the session, please. So it's our pleasure to continue the session with the talk from Mir Farzin Mashregi from the German Rheumatism Research Center. And he will talk about characterizing immune cells by single-cell sequencing. And we're very much looking forward to your talk.
How does it work? Okay.
And this one for the parent.
First of all, I would like to thank the organizers for giving me the opportunity to present our data on single-cell sequencing, and I would like to show you how powerful single-cell sequencing is to dissect, for example, in our case, chronic inflammatory diseases, and to find new ways to treat such immune disorders. So chronic inflammation is a very complex disease. It involves a lot of different immune cells, innate cells, but as well as adaptive immune cells, T and B cells. These cells are very central for driving chronic inflammation and maintaining the disease. And what is central is actually the interaction, the cooperation between autoantigen-specific T cells with B cells, which leads to the differentiation of so-called pathogenic plasma cells producing pathogenic antibodies, but also to T effector memory cells, which produce a lot of pro-inflammatory cytokines and chemokines, and therefore recruit pro-inflammatory macrophages, for example, and coordinate inflammation. But there is also another side of chronic inflammation. There are a lot of regulatory suppressive cells also present, which actually dampen the inflammation, like B cells producing IL-10 or IL-35 and suppress the inflammation, as well as T regulatory T cells, which help to control the disease. If you completely ablate the immune system and reset actually the immune system via autologous stem cell therapy, you can achieve progression-free survival in a lot of kind of autoimmune disease here, for example, for SLE, vasculitis, systemic sclerosis, MS, and so on. And this is actually the proof that immune reset works, that cure is achievable, and that imprinted immune cells are roadblocks to resetting immune tolerance in autoimmune disease. The problem with this kind of treatments is that you completely get rid of the protective immunity and therefore are very vulnerable to infections, and this hinders actually the widespread application of such kind of experimental therapies. So there is a medical need to identify cells which drive and maintain chronic inflammation, and to distinguish from those cells which are actually protective and help to resolve chronic inflammation. So because of time, I will actually today talk only about the central role of T helper effector cells in chronic inflammation, which, as I told, by producing chemokines and pro-inflammatory cytokines actually coordinate inflammation. So we took advantage of disease, which is the juvenile idiopathic arthritis. There is a lot of evidence that memory effector cells are massively involved in driving the pathogenesis of this disease. This is arthritis of onset before the age of 16 years. It affects approximately 1 in 1,000 children and has different subtypes. So we ask ourselves which T cells and how do these T cells contribute to juvenile idiopathic arthritis. So we took advantage of the single sequencing technology, and this has a lot of benefits actually. So by conventional methods, for example, if you dissect or investigate inflamed tissue, you get do by RNA sequencing, for example, you get actually the molecular identity of a mixture of healthy and diseased cells. And by doing so, you will might be able to identify an average clone, find a known drug, an immune suppressive drug, and treat the chronic inflammatory disease. And what you can see, you get you will deplete the average clone, but you still have other cells, maybe rare cells, which still contribute to the disease, and you achieve immune suppression, but not really cure. Whereas if you use single-cell RNA sequencing, you can identify different types of cells which contribute to the disease, and then design precision drugs to get maybe rid of all of them, or even to identify cells which are regulatory type and can be used for therapy, cell therapy. So what we did, we used paired peripheral blood mononuclear cells as well as the synovial fluid of affected children with juvenile idiopathic arthritis and isolated cytotoxic CD8 memory type T cells, effector T cells, as well as T regulatory T cells, and did single-cell RNA-seq using 10x technology platform and single-cell TCR sequencing to see to which specificity these T cells have. So this is a global overview. If you look at more than 70,000 memory T cells from these patients, and we were able to detect around 2,000 genes per cell and around 70,000 transcripts per cell. And you can easily see that the cells actually cluster here on the t-SNE map according to their tissue of origin, here from the synovial fluid, again here from the synovial fluid, and here from the blood, and according to different subsets of CD4 effector T cells, CD8 cytotoxic T cells, and FOXP3 positive T regulatory cells. So I will concentrate and zoom into the effector memory compartment. And if you look in more detail here, again on a t-SNE plot, you see that these cells are actually not homogeneous. You immediately see that you can distinguish 6 different clusters in these T cells. And this was very robust in the 7 patients we've analyzed so far. And if you look at the T cell receptor specificities and T cell receptors of these cells, you can immediately see this cluster number 1 here in orange and cluster number 3 in green, actually that you can see enriched clones. So significantly more T cells which were clonally expanded. And to investigate actually what these cells are, we performed a trajectory on this T cells isolated from the synovial fluid, and to see also the relations between these 2 interesting clusters, which show expanded clones, presumably autoantigen specific T cells. We see 4 nodes actually. And in order to find out what is the molecular identity of these cells and which functions these cells might contribute to chronic inflammation, we did a gene set enrichment analysis on single cell level. And what we could find that these cells from the right here, they had more resting memory signature, whereas the cells here on the left, the interesting ones, 1 and 3, showed a signature of in situ antigen activation. TCR signaling genes were induced in these cells. And one marker which gained our attention was actually that those 2 clusters expressed high levels of the surface receptor PD-1. And these cells, by the way, also showed a type of tissue residency. So this axis was activation, and we asked what is actually this action. Oh, excuse me, back. So we could identify actually in the green cells here, the cluster 3, actually suppressive genes expressed like IL-10, EOMIS, granzymes, whereas in the cluster 1 here, we could detect more actually CD40 ligand as a marker for antigen-specific antigen activation, TNF, CXCL13, and the cytokine GM-CSF. So this line, this axis is actually more inflammation. So here again, some genes depicted specifically expressed in pro-inflammatory cytokines on cluster 1 here, CXCL13, T-B cell interaction, interferon gamma, GM-CSF, and IL-21, which is important for the generation, for example, of pathogenic plasma cells. Whereas cluster number 3 here qualifies as IL-10 producing regulatory type 1 cells, which express IL-10, EOMIS, coenzymes, and so on. We were also able to identify several novel markers for these 2 clusters here, for example, CD39, a long non-coding RNA, which is specifically expressed by these pro-inflammatory cells. And for regulatory cluster 2, again, a non-coding RNA, interferon gamma, antisense 1, as well the novel marker NKG7. So we could validate these experiments. If you stain for CD39 and PD1, you can identify easily here that the CD39 positive ones represent the pro-inflammatory type of cells, whereas the CD39 negative ones express IL-10 and eomycin. One last data slide is that you can even in the blood, this is a t-SNE of the peripheral blood of this juvenile idiopathic arthritis patients, we observed a small cluster, very rare cells recirculating TR1-like cells from the SF within the blood, and they shared the clones actually with the synovial fluid of TR1 cells, showing that single-cell sequencing can be used for patient stratification and what's going on actually in the inflamed tissue. So here's already my summary slide. We were able to identify several types of different effector Th1 cells, for example, PD1 high T helper 1 type effector cells, ANAXA1 positive T effector cells, which we were never described before. We have to find out what kind of cells these are, and Th17 effector cells. And as you can immediately see, if you want to get rid of these cells, you need different markers to address therapeutically these cells. So this is a rationale for precision therapy addressing via multi-drug, addressing all these 3 effector cells. Whereas the biggest cluster here is actually bystander and derived from the blood. So they share clonalities, which we actually observed from cells from the blood. And the PD1 regulatory T helper cells, actually, we have novel markers which we can actually use to expand this population, and presumably these cells actually qualify as living drugs. So if you're interested for how we address to selectively ablate those cells, I would like to invite you to my poster and we can discuss this. So the work has been done by Patrick, and I would like also to acknowledge Pavel and Freddy's contribution to the bioinformatic analysis of the data generated on all the clinical staff who delivered the samples. Thank you very much.
Thank you very much. Are there questions? Yeah, please.
So actually, we took indeed patients which were treatment naive. So they got their first shot after we got the synovial fluid, actually simultaneously. So they were treatment naive. That's true, actually. We had a patient where we saw actually a depletion of the TR1 cells and an enrichment of the effector cells in one case so far. That's true.
There's another question over there, just behind you.
Can you briefly comment on the dynamics of these cell types? So I guess they will not be static, but rather dynamic. So changes of the subtypes from the one to the other or so.
So we actually did a lineage tracing according to the TCR specificities. And if you look at this, they are actually sharing a lot of clones, which we also find back in PD1. And if you remember the trajectory, they feed those cells here, so they are not activated yet. So, excuse me, they are not activated yet, and if they expand in this line actually, whereas the regulatory type PD-1 cells, there were no overlap, no significant overlap between the other classes. So we think they might come from the blood, but this analysis has to be finished first.
Thank you very much. We unfortunately have to move onwards. Thank you for your talk.
So we invite now. No. So we invite.
We have now a special way of presentation. I have been informed that the next talk is going to be given as a Skype talk. I don't know how it is played in. And if it works, then we have as next speaker Maxim Mamonkin from the Baylor College of Medicine in the US on engineering T cells to target malignant and pathogenic T cells.
Great, thank you very much. And I would like to thank the organizers for inviting me, and I apologize for not being able to fly down to Berlin. So I'll try to give this talk as best as I can. Hope you guys can hear me, otherwise please let me know. So I'd like to share a story how we can engineer cell therapies against T cell malignancies and also other pathogenic T cells. And so just to briefly make sure we're on the same page, we'll be using chimeric antigen receptors here. And CARs are the engineered molecules that enable T cells to directly recognize tumor antigens on the cell surface through an antibody-derived targeting moiety, which then transmits signal and activates T cells. And we can use this to essentially redirect patients' own T cells against cancer. For example, if a patient has a B-cell malignancy that expresses CD19, we can put a CD19-specific CAR through viral or non-viral vectors and then expand the cells and then give it back to the patients. So they will track down to the tumor eliminated. And so the goal of my lab and the overall question is, and challenge, is to extend this potency to more challenging diseases like those that are themselves of T cell origin. So the question is, can we actually engineer CAR T cells to target T cell antigens, those that are expressed on CAR T cells themselves? And so if we can solve this problem, this allows us to try to target T cell malignancies such as leukemia and lymphoma, as well as possibly develop therapeutic strategies for autoimmune complications that are driven by pathogenic T cells, such as immune rejection and graft-versus-host disease. But obviously, before that, we have to solve two main problems. One is, how do we avoid self-targeting of CAR T cells, because these T cells will essentially recognize these antigens on themselves and kill each other. And if we solve this, then these T cells upon infusion can also attack normal T cells in circulation and potentially cause a dangerous T cell aplasia, as we all know. It may be quite devastating long term. And so today I'll be showing you two directions, a few stories right there, how we can engineer these T cells against T cell malignancies, lymphoma and leukemia. And I'll be primarily focusing on unpublished data and showing some clinical results here as well. And then in the last part of my talk, I will try to convince you that we can also use this approach to extend it to non-malignant indications. And so first, I'll focus on the T cell malignancies and show how we can solve CAR T cell fratricide and T cell aplasia. And patients with relapsed and refractory T cell malignancies are really in dire need for effective therapies, because those few options that exist for them are very toxic, not always effective. And so if one wants to come up with a cell therapy that would benefit as many T cell patients as possible, one has to target antigen that are broadly expressed in T cells, so-called pan-T cell antigens that are listed here in this table. And so we started with CD5, which is very widely expressed in ALL and also various subtypes of T cell lymphomas, and its normal expression is pretty much limited to T cells and a small subset of B cells. So it's been previously targeted by an antibody conjugated with immunotoxin, but without any strong efficiencies right there. So we made a CD5 specific CAR based on that, one of those clinical antibodies, and then I made a second generation CAR with CD28 co-stimulation. And so obviously you would predict that once you express this CAR in T cells, 100% of which express CD5, they will just simply kill each other. And we thought so too, but in fact, what happens is quite unexpected. When we put a CD5 CAR on T cells, we immediately found that these T cells downregulated CD5 by flow on the cell surface very, very rapidly right after transduction. So you can see here, control T cells are CD5 bright, but CD5 CAR T cells actually downregulated. And it's not just masking, because we also don't see this protein by Western blot. And so it really is internalized and degraded, even though the mRNA production is still intact. And so this interesting mechanism protects these T cells from fratricide and enables very efficient expansion after transduction. There's initial confusion, initial fratricide after transduction, but ultimately they're able to overcome and expand. And so once they expand, they are actually very cytotoxic against CD5 malignant T cells. Shown in an example here when we co-cultured them with jerkets that naturally express CD5, I should say UCAT. Those cells are very rapidly eliminated upon co-culture with CAR T cells, as opposed to control cells that don't really touch them. And we also show that they're effective in mouse models of disseminated ALL and produce elimination of tumor, at least initially, and produce a survival advantage using two very aggressive models of systemic ALL. And this is all published data. So this has led to the development of a clinical study at Baylor that we call Magenta protocol, which is open to all patients with refractory relapsed T-cell malignancies that essentially have exhausted their therapeutic options and retain CD5 expression on malignant cells. This is a first-in-man study, so the toxicity profile is unknown. That's why we built it as a bridge to transplant. This is a dose escalation study where patients receive a single infusion of CAR T cells following lymphodepletion, and the trial overview is shown on the bottom. And a few words about manufacturing the cells in the GMP that we have right there at Baylor, one floor below. We take peripheral blood for those patients, we freeze it down, then we thaw and transduce it with a retroviral vector containing CD5 CAR 3 days later, then expand in G-Rexes in the presence of cytokines, and then cryopreserve. And so the standard manufacturing protocol mandated cryopreservation day 7, which will later amend it to allow preservation earlier between day 4 and 5 post-transduction. And this is called a short manufacturing, and this will become important a bit later. There's no problem manufacturing these cell products for most patients. Fratricide is never an issue. We test them very rigorously before infusion, and specifically we test for the presence of residual malignant cells that may contaminate the initial starting material. And more than 90% of cell products actually meet the release criterion. So what we see in patients is that predictably CD5 CAR T cells expand and persist quite transiently. So this is very typical for CD28 zeta CARs. And we don't really see any overt toxicities, no severe CRS, only just mild CRS and mild neurotoxin in a couple of patients, no infectious complications, and no non-hematologic adverse events. So typical profile that is also such typical for other CARs that we see and all other centers are testing. So is this because these CAR T cells don't work? Well, I wouldn't say so, because actually we do see an evidence of clinical activity in quite a few patients. So this is a gentleman with very disseminated PTCL. You can see a large chunky tumor burden throughout, a lot of PET-avid lesions throughout the body. And he was resistant to several lines of therapy. And then upon receiving a single dose of CD5 CAR T cells, went into complete remission. So these T cells are actually quite cytotoxic in patients. So this is a brief overview of current results so far. About 50% of patients with T-cell lymphoma ultimately undergo complete remission, and they have various diseases ranging from non-otherwise specified peripheral T-cell lymphoma, also angioimmunoblastic and Sézary. And things are noted as rosy in T-ALL patients. Unfortunately, we did see transient stabilization of disease in a couple of patients on the CR and one, but it's not as robust as in patients with T-cell lymphoma. And interestingly, if you stratify these patients by manufacturing protocol, you can see very clearly here that the patients that receive cells frozen later on 7 days post-transduction never see any, never produce any complete remissions, whereas all the responses that we see are only observed in patients that receive a product that was manufactured with a shortened protocol. And specifically in the lymphoma patient, 3 out of 4 of them went to complete remission. So this kind of highlights the importance of optimal manufacturing of the cells, potentiating the responses. What about endogenous T cells? So one interesting thing that we observed in this trial is that we never see selective ablation of endogenous CD5 CAR T cells. So this is an example of one patient that underwent complete remission, and you can see that this is the shaded area indicating expansion and contraction of transgenic CD5 CAR T cells. And by the time they're gone from peripheral blood, we still see quite a few endogenous CD3 positive cells that contain both 4s and 8s, and eventually they expand. And these cells retain CD5 expression. In fact, when we looked at all the patients, the minimum number of T cells that we see probably at the peak of response is never 0. It's always between 7 and 30%. So unlike patients with B-cell malignancies, in which B-cell aplasia is very common, we don't really see ablation of endogenous T cells, even in patients in which T-cell malignancies melt away. So just to quickly sum up this part, we are seeing that the CAR T cells, C5 CAR T cells, are safe, at least at current dose levels. They have strong curative potential, particularly in T-cell lymphoma. We still have a lot of work to do, especially in T-cell ALL. And an interesting thing, at least in these dose levels, we do not see ablation of endogenous T cells. So currently we're trying to potentiate CAR T cell activity by escalating the dose and also introducing some other manufacturing changes that are designed to improve the potency of them. And another way to improve the outcomes in patients with T-cell malignancies is to explore other targets, such as CD7, which was also in that original table. It's very widely expressed, particularly in T-cell ALL. And so we thought that maybe we can use the same trick that we used with the CD5 and just express CD7 specific CAR on T cells using exactly the same backbone that's used in the clinical grade CD5 CAR. But no, that unfortunately didn't work. And as you can see here, there's still a lot of residual expression of CD7 on the CD7 CAR T cells after transduction. And as a result, the cells essentially just chase each other down and eliminate, and the expansion is very poor. Now, we can help them to get rid of CD7 by CRISPRing it out in activated T cells, and this produces very effective knockouts. Initially 90%, now we've optimized it up to 97, 98%. And then after we transduce it with a CAR, they are able to expand very nicely as they become resistant to fratricide. Again, this work was published before, so I'm not going to emphasize it that much. I'm just going to say that it just introduces one extra step in manufacturing tumor-specific T cells by CRISPRing it out first before we could put a CD7 CAR in. And then the CD7 CAR T cells expressed on CD7 edited T cells expand and exhibit very potent anti-leukemic activity upon co-culture with primary malignant T cells isolated from patients at Texas Children's Hospital here. And so within 24 hours, they just eliminate them, and also very protective in aggressive models of systemic ALL. We are very excited to start a clinical trial very soon that's based on this study. This, again, will be a dose escalation bridge to transplant study as a first in men, and then it should begin in a few months at Baylor as one of the first CRISPR-edited CAR T-cell trials, at least on this side of the pond. So in the second part of my talk, I wanted to briefly focus on the other parts of this work. So by now, we've shown that CD5 and CD7 CAR T cells can effectively target T-cell lymphoma. And can we use this knowledge to extend it to pathogenic non-malignant T cells that are devastating in certain conditions like auto and alloimmunity? And so particularly focusing on the model of allogeneic immune rejection. And we don't have to go very far because this problem also exists in adoptive cell therapy of cancer, as the field gradually moves from autologous cells towards banked off-the-shelf CAR T cells, which may resolve a lot of problems associated with autologous products, such as very impotency and also the necessity to produce them specifically from each patient, which complicates the manufacturing, makes it much more expensive. If we pre-manufacture the cells from healthy donors and bank a lot of them and then just administer as needed to patients with suitable disease, this can significantly reduce the cost of manufacturing and unify and make the potency more uniform, and make them available off the shelf. So we don't have to wait until the product is made. However, this approach is usually associated with two very important limitations, such as graft-versus-host disease that the T cells may be caused, and also host versus graft, where the host, the patient, him or herself, may recognize the adoptively transferred cells. And so the first problem is relatively easy to overcome. We can disarm these T cells by removing TCR or using T cells with defined specificity, and this pretty much gets rid of the graft-versus-host problem in the setting. But the alloimmune rejection of the host becomes a very difficult problem to solve. And in fact, when we model this in vitro, when we mix peripheral blood mononuclear cells from one donor with T cells from another donor, we see that these T cells are rapidly rejected within 9 days. And again, there's no news here. This has been known for decades. So we can probably make them invisible to T cells by removing MHC class 1 and class 2, which would impair T cell recognition. But what it will do is will aggravate NK cells and activate their NK cell mediated rejection. In fact, that's what we see when we mix NK cells with MHC class 1 knockout donor T cells. Now rejection happens much faster because NK cells don't really need any time for priming. They are already primed to kill, and within 2 days, they just eliminate those T cells. So this probably would require very elaborate engineering and possibly host conditioning with continuous immunosuppression to achieve potent anti-tumor activity, which is obviously very suboptimal. So we decided to tackle this this question and this problem from a different angle. So we know that T cells, upon recognition of alloimmune or autoimmune autologous antigens, they become activated, and so they upregulate the expression of activation markers. So we T cell biologists know them by heart now. One of the most prominent is 4-1BB. A lot of immunology and tumor immunology folks are using it to define activated T cells, and also other markers are listed here. And once they are done eliminating targets, they go back to resting memory cells that downregulate these markers. So we thought, what if we engineer an additional receptor that we can express on our therapeutic cytotoxic cells that would essentially recognize these pathogenic T cells, recognize activation markers on these pathogenic T cells, and selectively eliminate this population without touching all the other non-alloreactive cells. And so we call this receptor alloimmune defense receptor, or ADR. And so that's what we did. We engineered this ADR. We put a 4-1BB target in the domain, initially from the 4-1BB ligand itself, and then as derived from the antibody. And then we conjugated with the first generation CAR, essentially the CD3 zeta, and then also fused with GFP to enable tracking the cells. And you can see that ADR is efficiently expressed on the cell surface of transduced cells. And once we put them on T cells, the expression of ADR does not impair their expansion. They expand just fine, very comparable to the control transduced cells. So now, how does it work? Will it enable selective recognition? So we co-cultured the ADR T cells with autologous, freshly isolated T and NK cells from PBMCs. And so you can see that there's absolutely no activity against resting cells that are fresh from the blood, and also no activity against NK cells. But when we pre-activated these T and NK cells for 48 hours, we start seeing selective targeting of these populations when co-culture with the ADR cells. And this happens for both T and NK cells, as you can see here. So will this help prevent T and NK cell mediated rejection? So we used a very old assay established in the immunology field decades ago, called mixed lymphocyte reaction, or MLR. In this assay, we mix T cells from one donor, in this case it's a HLA-A2 positive donor, with PBMCs from another donor, in this case A2 negative, one at about a 1 to 10 effector to target ratio, target to effector, I should say. And then in this model, the PBMCs start recognizing the donor T cells that have been disarmed, again, by removing TCR, so they don't fire back at them. And then within 3 to 6 days, we see the onset of rejection. And this rejection is completed within 9 to 12 days, as you can see here. But when we expressed ADR on those cells, you can see that the cells resist rejection and actually expand quite a bit. And they don't just eliminate all T cells, the host T cells still exist. They still are present in the co-culture. So essentially, they just selectively ablating activated cells without touching naive ones. Then we can model NK cell mediated rejection.
Maxime, could you come to that time is over? So if you could sort of already come to the conclusions.
I thought I have 20 minutes. No. Okay. Well, I can briefly summarize it now. So basically, what we show is that the cells are protective against rejection in vivo, and then when we combine them with a CAR, they are able to expand and persist in mice that have the pre-activated T cells from another donor, and they persist and eradicate leukemia, unlike mice that receive unarmed CD19 CAR T cells, in which this persistence is abbreviated in the mice, and the leukemia relapses very quickly. So we think that this is an interesting way to protect off-the-shelf T cells from immune rejection in patients, and then still retain their anti-tumor function. And so I will quickly end here and acknowledge people who did the work in my lab, as well as CGT leadership, funding sources, obviously patients and their families. And we're looking for very talented postdocs who could take on new ideas. Thank you very much. I'll take your questions.
Thank you very much for a very impressive talk. Questions? Dieter?
Thank you, Maxim, for this nice talk. I have one question to the second or to the last approach. I think the problem is here that the allo-activated regulatory T cells express as well 4-1BB. That means you kill both the good guys and the bad guys. What do you think about it?
Yeah, exactly. And we think that's not a problem in the cancer setting because Tregs you mean the regulatory cells that express 4-1BB, right? So it's not, we don't think that targeting Tregs in the cancer setting, at least temporarily, would be a problem. It may be a problem in autoimmune diseases, yes, and that we are developing other ADR targets apart from 4-1BB that are not targeting Tregs, but at least 4-1BB specific cell ADRs I think will be useful in the cancer setting.
There's another question.
I also have a question to the last part. So would that mean that any activated T cell in future would be eliminated if your CAR T cells with the ADR are persistent?
Yes, that's a great question. So obviously ADRs will not be able to tell whether the responses are detrimental or productive, such as those that are against pathogens. So we did see that ADRs can suppress sort of in vitro the activity of virus-specific T cells, although we never see suppression of the initial round of killing. So the virus-specific T cells or pathogen-specific T cells, they're already kind of pre-primed there. So ADRs do not ablate them completely, but they may eventually target them. We think that's not going to be a problem long-term because eventually the cells will be gone from circulation, and then they have to really be in the same tissue, in the same organ to suppress this activation. And clinical studies also with other potential ADR targets like CD30, which is also expressed on activated T cells, show that there's actually no suppression of immunity and when targeting these antigens. So I guess we will see in the upcoming clinical studies, and we'll go from there. But at least if we start seeing some detrimental effects, we show that we can express ADRs on virus-specific T cells themselves, and they are able to achieve both anti-leukemic, anti-rejection, and antiviral function.
Thank you very much. And I'm afraid we need to move on and like to hand over to the next chairs. Thank you very much.
All right, thanks.
So in the name of two of us, of Ali Turhan and Uwe Marx, myself, I would like to invite you to the second session. This is a preclinical session. We are discussing preclinical models, and as you know, the preclinical models and the predictivity of preclinical models is a major issue for the development of these advanced therapies. It's difficult to have human living cells and test them in animals. Therefore, we are focusing on in vitro systems. The session starts with Antonella Motto, an associate professor at the Department of Industrial Engineering, University of Trento in Italy. And it's my pleasure to invite you to the scene, Antonella. The title is Engineered Polymeric Matrices as Instructive Cells, Artificial Microenvironment. Antonella, the floor is yours.
Thank you very much for your kind introduction. And first of all, I would like to thank the organizer for inviting me, giving the possibility to share with you part of our work we are doing at the Biotech Research Center at the University of Trento. We had already excellent example of system that should be used in regenerative medicine and tissue engineering in the previous talks, but I would like to focus my presentation on strategies to design polymers that can be used to fabricate this kind of matrices. The idea in our lab, considering that the scaffold should be used in combination with the cells, in combination with the cells taken from the patient biopsy, or more in an easier way can be used to recruit the patient cell direct into the body, so they don't have to be preloaded with the cells, just to simplify the system. Polymers can be used as they are, but nowadays it's much better to recapitulate some aspect of the biological system, that means basically the extracellular matrix, into the polymer by conjugating some peptides in order to have an improved but also specific bioactivity into the polymers. The polymers can be used then to encapsulate the cells, or can be fabricated and then seeded and populated by the cells. One of the strategies in this framework is a biomimetic approach, is the use of nature-derived polymers, because are designed by nature, because now we are able to design fabrication methods almost standardized and simplify, low cost, and in mild conditions, can be blended with other nature-derived polymer or synthetic polymer, can be functionalized, can be combined with the drug release systems. One example that I would like to start with is alginate. Alginate is a polysaccharide isolated from brown algae, and it is largely used because it's possible to obtain beautiful hydrogels in very mild condition, cross-linking with the calcium chloride, following the scheme you have in your slide, and providing some functional groups that can be used to decorate this kind of polysaccharide. Why decoration? Actually, the problem of alginate is that it's not really bioactive, or not specifically bioactive. So this is just a piece of work done in collaboration with Dr. Yaudanskaya. The idea was to see how this bioactivity can be improved in the first step by blending alginate with other proteins. We organized and designed a co-culture in vitro experiment in order to have an idea about the impact of the system on angiogenetic factor. So we decided to fabricate hydrogels by using just alginate or mixing alginate with a protein isolated from silk that I will introduce in a couple of slides, or alginate in combination with the collagen. As you can see in the right part of the slide, when cells are entrapped into the alginate alone, they can survive quite well, but they are not really active and the proliferation is a little slow. But when some other protein or factor are included, for example, collagen, what was observed that there was the proliferation was upregulated. But interestingly, when the silk fibroin was introduced into the system, cells, in particular endothelial cells co-cultured with mesenchymal stem cells, were able not just to proliferate, but also to assemble in capillary-like structure. Just recently, we tried to move on, considering alginate as a possible polymer that can be used for organ printing or for hydrogels, to decorate this kind of chain by selecting specific peptide depending on the medical problem we want to face too, and by designing chemical reaction that are into the green chemistry or click chemistry in order to have modification of the alginate just by using RGD containing peptide, or we can select other peptides and we can decorate and we can build up a specific protein for a specific application we want to face to. But in our group, we have quite large experience in the use of silk-derived protein. Actually, silk has a long story into the biomedical field, starting more than 300 years ago when Dr. Paré started to use a silk filament as a filament ready to be used for suture, in particular in the vascular suture. But then by the time we are learning how to process, not just to use a filament produced by nature, but by dissolving these proteins, I will show you in the next slide, and by using the solution, we can build up a new structure that doesn't exist in nature, but by using natural protein. You have some example just to give you some ideas. We can produce sponges, we can produce hydrogel with different properties, or fiber again. Actually, silk-based devices are already into the market, FDA approved. There is a company in the States, just to mention one name, Allergan, that is producing a scaffold for tissue and ligament and tendon regeneration based on silk. And in Korea, they have other approved material for cornea regeneration as well. About the process, that is a quite interesting protein because we always start from the cocoons. Cocoons are produced in well-controlled environment. It's a kind of medical grade material. It's reproducible, can be standardized. So we have a quality control from the beginning. This cocoon can be purified and one protein can be isolated. The protein can be then dissolved. So we start almost always from a water protein solution without any chemicals inside. So we define different kind of methods to fabricate devices. As you can see, we have sponges. We can produce sponges, but mainly hydrogels that are very interesting into the field to design artificial matrices, can be in combination with synthetic polymer like a PVA for this kind of wound healing device, or in combination with the hyaluronic acid for cornea regeneration, or just recently we developed a new chemical modified fibroin that can be used for 3D printing. And this structure is quite interesting because it can be produced, can be squeezed, aspirated with the tip of your pipette, and then released again, and the material is able to go back to the same size and the same shape. What is interesting in fibrin that should be underlined is a protein. So depending on the process, what we do basically is to change, first of all, the secondary conformation. By changing the secondary conformation, we change the arrangement, the 3D arrangement of the molecules and its interaction with the water, so affecting not just mechanical properties, but also the degradation rate, as well as the biological performances. So we can tune, sometimes quite often, the the biological activity, just changing the secondary structure of the protein by selecting specific processing methods. Just an example of what I'm saying, this is a work done in collaboration with a group of Colorado University at Boulder, and the idea was to produce hydrogel by using different methods, but just the protein plus water. Depending on the method, we can obtain material from very creamy consistency to very compact and elastic, and what was changed basically is just the conformation of the protein. So for this work, we were interested to see how we can have an impact on stem cell fate, just changing the mechanical properties of the hydrogel. So the hydrogel was produced by changing the protein concentration, and human mesenchymal stem cells were used, and then we were interested to see if the system were able to differentiate in vascular smooth muscle cells. And as you can see, just by selecting these mechanical parameters, we were able to obtain this kind of differentiation without changing the culture condition, but just tuning the mechanical properties of the substrate. Other hydrogel can be prepared in very simple, with simple methods. This is the method named salt leaching, just mixing the water solution with sodium chloride crystals, and the protein goes into gelation very fast, and then the salt can be washed away, so obtain this kind of hydrogel porous material. This material, of course, can be used as it is or can be combined with other biological structure. For example, can be preloaded with the stem cells or can be included and can combined with a sulfur donor. I will explain you the why of this strategy. First of all, in collaboration with the medical school in Modena, we were interested to see if the sponges made by fibrin were also osteoinductive. We were already observed in vivo that they were able to generate new bone, but the idea was to have an idea about the osteoinduction. So the protein was fabricated, the scaffold was fabricated with the salt leaching method. You have a porous sponge, and as a control system, we use a sponge made by polylactic acid. Both sponges were implanted subcutaneously into the rat in ectopic implant, of course, and after 40 days, we control the situation. Of course, at the same time, we implant also the same samples, but preloaded with dental pulp stem cells and amniotic fluid stem cells, just to compare the results. If you look at the first line, when the two materials were implanted alone, no bone was observed into the histological section, no mineralization. But when the cells were included into the fibrin silk system, you can see very well through this alizanin red staining that this ECM was mineralized. No data, no effect when the cells were mixed with polylactic acid. This is because it's not enough to include the stem cells, but the stem cells, when they are included in an artificial matrix, they should have a kind of crosstalk, positive interaction with this material to have some result. Another strategy was designed in collaboration with Orthopedic Institute Rizzoli in Bologna, Italy. We start from the observation of endogenous production in our body of sulfur donor at nanomoles level. That is able to upregulate the osteoblast activity and to downregulate the osteoclast activity at the same time, and pushing also stem cells to become osteoblasts. Why this strategy? Because the idea was to simplify the system. Can we avoid to include the stem cells into the scaffold, but just a drug system releasing in order to push our body to produce what is necessary to produce? So we identified this molecule. The name is GYY, and this work was done in collaboration with the Emory School in Atlanta. And they observed that when this molecule was injected systematically into the rats, even in the osteoporotic model, a upregulation of the bone regeneration was observed. But the limitation was that this molecule should be injected every day. So we started to discuss how can we solve the problem. So we decided to combine this GYY with our fibrin sponge. And as you can see, we select as well the salt leached sponges. In this case, to have a system that can be implanted with a single administration of GYY, and GYY is released just in the peri-implant site. So the material was, the system was evaluated in vitro by using bone marrow-derived human mesenchymal stem cells. And then after 20 days, we checked some marker that can give us an idea about the biological performances. As you can see, just after 7 days, it was observed that the production of VEGF. This is quite important to have a very early angiogenesis. But after 21st days, all the markers related to bone were improved, upregulated, like alkaline phosphatase and calcium deposition, as well phosphate carbonate deposition. Both of them are the natural component of the natural hydroxyapatite. But the question was why this impact? Because we observe, first of all, that the GYY is released for the first day and then disappears. But nevertheless, we observe data improvement after 3 weeks. So we check some skeletal development, gene expression on this marker that in all cases were upregulated or anticipated when GYY was into the system. And also the osteogenesis was upregulated at a genetic level and osteoblast activity was inhibited. But then what was interesting also is to observe the integrins gene expression. And as you can see, what is particularly interesting, I would like to focus your attention on this ITGA2 that is integrin involved into the catenin pathway. That is a pathway that can trigger the endogenous production of sulfur donor. So what we did was just the initial input, leaving the nature to do the job as nature can do. Another example how we can modulate the stem cell fate or activity was by using another androgen. In this case, it was chemically cross-linked by using genepine, and the blue is due to the reaction between genepine and the protein. And then we see the cells, mouse embryonic stem cells in neurogenic media, and we compare the behavior with the cell seeded on TCP. As you can see in TCP, the cells were able to not just to proliferate, but also differentiate. But when seeded on top of this androgen, they were not able to differentiate, maintaining the pluripotency, but just proliferating in cluster. So we decided to change a little bit. You can see we check also the early marker for differentiation and beta-3 tubulin. You can see always was downregulated when cells are in contact with this gel. But just changing the fabrication method, moving from chemical cross-linking to physical cross-linking by using ultrasonication, so inducing different conformation in the protein, we repeated the same experiment, having a completely different results, where cells were able in 2 weeks to differentiate, as you can see through the MAP2 that is a mature differentiation marker. One step more, why don't decorate the fibrin with something else? This something else, we decide to use the peptide, the laminin-derived IKVAV, and this peptide was chemically cross-linked with the basic protein chemistry to the protein. Again, the same hydrogel was produced, and then we observed again that we had an improvement in speeding up of the process. As you can see, comparing the unmodified protein with the peptide modified protein, you can see that all the markers were improved with this functionalization. What is interesting to observe that in unmodified but even modified material, the GFAP was not observed. This is quite good signal because they are responsible for scar tissue formation in the scar. But then we move to the other question, what about angiogenesis? So we co-culture outflow endothelial cells with the human mesenchymal stem cells on both matrices, unmodified, modified. And as you can see, after 10 days, in both cases, endothelial cells were able to proliferate and to assemble in very capillary-like structure, well-branched in both. That is a kind of this data can confirm the intrinsic property of the fibrin that is able to upregulate the angiogenesis into the system. The last example I would like to provide you before the end of my talk is another one that is work done in collaboration with a group of Professor Martin von Griesben, at that time at Technical University in Munich in Germany. The idea is, okay, we can play with the morphology, we can play with the mechanical properties, we can play with the functionalization. What about to put all together? So we decided to design a scaffold with a morphological gradient, with isotropic transition zone and isotropic for ligament regeneration, and so obtaining an isotropic area. So we have a kind of morphological signal to the cells, and then this sponge was functionalized, first of all, with the heparin as a bridge, and then the anisotropic area with TGF-beta2 and isotropic area with GDF5. So we obtain a system able to maintain this peptide stable into the matrix, and then again, an in vitro test was performed, and as you can see, this is a not modified. This is the area with just TGF-beta, just GDF5, and both growth factors. And you can see that when we have a combination of the morphology gradient plus the interaction between the two factors, even if they are cross-linked to different areas of the scaffold, we had the improvement on all the markers in the specific area. That means collagen type 1, you see in more anisotropic, collagen type 2 in the transition zone, and the collagen type 3 in the other area. This is a quite interesting results. Of course, we are still working on to have a more detail on this mechanism. So just to conclude and to summarize the concept, we are looking for new strategy for fabrication method that can take into consideration some cues, biological cues that should be recapitulated, to design system that can be better controlled, simplified because better control, with the standardized fabrication methods that can also have a kind of low cost and easier transition to clinic and to the market for the regulatory aspect for the approval. To conclude, this is our group, biotech group. This photo was taken during the last Frontier Seek Technology that was organized in Trento in June this year. I would like to acknowledge the REMIX project that we are running at the moment. It is based on the use of natural polymer for biomedical application, in particular regenerative medicine, tissue engineering, and of course expert tissue, where most of the work was started from. But also, I'm quite proud to announce that a new Gordon Conference series was recently approved and will be on silk science and technology, from textile, biomaterial, tissue, and behind. So all kind of application. And the first edition will be in New England in 2021. I have a pleasure to be co-organizer with Professor David Kaplan of Tufts University. And of course, thank you for your kind attention.
Thank you. Thank you, Antonella. Thank you very much for that presentation. The talk is open for questions and for discussion. Then I would like to start. Do we have someone there?
Yeah, hello. Thank you for the talk. Here I am.
Okay.
Anyway, I can hear you.
You hear me. I have the question regarding you nicely showed that you can adapt or modify the features of your material, maybe the silk, by adding something in and my question is, because it's important for the acceptance in the body that you have a good immune regulatory response, that means from macrophages. Did you also try if your changes or modifications are able to also impact those remodeling features by maybe M1, M2 type macrophages?
Yeah, that's a very good question. Actually, we have already had some data on that. The work we did in collaboration with Professor James Kirkpatrick, we implant our material in the animal model. We observed that the fibrin-based material were able to speed up the transition between macrophages type 1 to macrophages type 2 by nature, intrinsically. Now we are just started to work in a specific silk modification in order to better drive this kind of transition, because this is the key. We can control other phenotypes, but we should start from the immune system and macrophages work or performances, in particular the transitions. Yeah, we are working this, but it seems that the key point are the peptide released by the fiber during the in vivo remodeling. This peptide, it seems it has an effect on macrophages type 2 transition and activity. Yeah, that is a very good point.
I do have another second question. All these material silk-based or alginate systems are non-human systems, and they made a great job for the last 40 years. If your perspectives from your point of view, if you look 10 years above, do you think that transplantable materials can be on human origin only, and can we use these human origins also for preclinical modeling of human organs, or do you think we still will stay with xenogenic?
So if I understood where your question is on if this if silk-based material can be implanted depending on immune acceptance. Yeah, actually there are data on that, and there are no problem related to, because the protein is a very simple composition, because the 95% of the amino acid are 4 amino acids. And it seems that our body can recognize, and there is no negative impact on this. What was the problem related to silk, the social filament, because it's too crystalline. So the idea is to define a fabrication method that can reduce beta sheet crystalline phase to improve the alpha helices, different interaction with the protein. In this case, there are no problem related to immune acceptance. I think it can be, there are products already on the market in the States.
Thank you. So with that, thank you again, Antonella. And we come to our next speaker, Manuela Gomez, Associate Professor of the University of Minho, and she will talk about the stimuli responsive and multifunctional scaffold cell systems for tendon tissue regeneration. Manuela.
Okay. Good morning, everybody. Thank you very much for the presentation, and thanks the organizers for the opportunity to show us today some of our work on the development of tendon tissue engineering strategies that based on stimuli responsive and multifunctional materials targeting tendon regeneration. So, as you might know, tendon injuries and pathologies are highly prevalent. So, in fact, they represent 30 million related procedures that are performed annually, and this is only to solve the most critical situations. And in fact, they represent 50% of all the musculoskeletal injuries. And the problem is that this tissue has acquired low healing and regenerative ability, mainly due to the scarce number of cells that compose this tissue, and also to a low vascular supply, which means that with currently available techniques, including the surgical techniques that usually involve either suturing techniques or using graft materials, they do not restore the initial characteristics of this tissue. And this is the main problem, because it involves the high risk of re-injury or of chronic pain to the patients. Furthermore, these injuries are growing due to the aging of the population that has an increased risk of undergoing this type of injury, and also in active population and people that are practicing more exercise. So we believe that tissue engineering may offer the prospects of achieving therapies where we can target tendon regeneration instead of this healing that does not restore the initial properties of the tissue. So, but in fact, tissue engineering strategies need to address several issues for tendon regeneration. So the type of cells that we are going to use, the biomaterials, and the mechanical stimulation. And we need to address all the specificities of the different tendons that are located in different anatomical regions of our body and that therefore have their unique characteristics and unique requirements that we need to target. Nevertheless, in tendon tissue engineering, we definitely need to develop a stimuli-responsive construct that has a biomimetic architecture, and this represents actually a number of unmet requirements with the currently available techniques in for tendon tissue engineering. And this is mostly because, as I said, it is very important to mimic this structure of tendon that is quite unique and is responsible for its remarkable properties that have to withstand high mechanical loading. And this is a structure that is composed of a hierarchical fibers from the nanoscale to the macro scale in a typical arrangement and alignment that is responsible for its quite unique mechanical performance that we need to address as well. The other important issue, as I said, is also the cellular contact. As we know, tendon have a scarce number of cells, so we need to study other cell sources that we can transform in cells that can regenerate this tissue and form this tissue. And we need also to consider their interactions with underlying and produce matrix that is under constant loading. This is also a very important factor. And another challenge in tendon regeneration is also how to find tools that can help us to somehow modulate the inflammatory response, because the healing mechanism in tendon is never successfully achieved, and that's why in most cases it is formed an unorganized matrix that has not the characteristics of the initial tissue, and therefore we need additional tools to prevent the formation of the scar tissue that is usually formed when this process is not complete. Moreover, there is not so much knowledge on the process involved in tendon development, healing, and repair, and this has been hindering also the development of novel and better strategies. Nevertheless, it's well known that mechanical forces play a fundamental role. Although the mechanisms are not totally known, we know that there are numerous intercellular and cellular interacting signaling pathways that regulate the most important process involved in tendon healing, development, and differentiation. And for all these reasons, we have been focusing on the use of magnetic actuated strategies for tendon regeneration, and these involve basically the use of a tissue engineered magnetic responsive construct. And we focus on these strategies for several reasons. First, because magnetic stimulus provides way to apply mechanical stimulus through a contact-free technology. It can be remotely actuated both in vitro to induce a specific cell response, but also upon implantation of the construct. Moreover, magnetic stimulus has been shown to play a role in the modulation of inflammatory response that we consider a key issue to achieve tendon regeneration instead of the mere healing. And these magnetic constructs can be obtained by two different ways. One is to magnetically label the cells that can actually with magnetic nanoparticles that can actually be tagged with different entities and provide different functionalities, and then see them onto a 3D construct. Or we can develop a magnetic construct where we then see the cells, and in either case this can be then actuated, as I said, both in vitro and in vivo through the application of an external magnetic field. So for this reason, we have been developing many different approaches based on such strategies, and also because we need different strategies, as I mentioned earlier, to target the different tendons that we have in our body that have different requirements, and with the two main goals. One, to obtain the tendinogenic differentiation of stem cells, and in particular stem cells from adipose tissue, and another that is how to modulate the inflammatory response. So we have first focusing on the stem cell source that we should use for achieving tenocytes, the cells that form tendon, and we have focused primarily on amniotic fluid stem cells and adipose-derived stem cells. And they seem to respond differently to different growth factors or to different biochemical stimulation that we provided in the medium, although they were not seen to demonstrate a robust tenogenic differentiation. And we went also to study, for instance, subsets of cells within the adipose-derived stem cells that could be more prone to tenogenic differentiation. And we end up using these cells that are positive for tenomodulin and that seem to be more prone to tenogenic differentiation than the unsorted cells. We culture these with the different growth factors, and here in particular TGF-beta3 and GDF5 that have been reported to induce tenogenic differentiation. And actually we have seen in this case an upregulation of the expression of scleraxis, that is one of the most specific tendon markers. But nevertheless, when we went to see at a more protein level, although TGF-beta3 was found to be a significant and potent inducer of tenogenic differentiation, it was not enough to trigger the downstream moleculars representative of the tendon mature phenotype. And this showed us that actually, considering that tendon is such a mechanoactive tissue, we might need to combine other factors like mechanical stimulations besides the biochemical stimulation in order to mimic better the characteristics of the tissue in vivo to obtain a more robust tenogenic differentiation. So nevertheless, one of the strategies that we first pursued was to develop of these magnetic responsive adipose-derived stem cell sheets. They were actually built using the subpopulation that I just mentioned, cultured with these magnetic beads. And with these very simple strategies, and as we use magnetic stimulation for stimulating matrix production, after 7 days of culture with these particles, we obtain a very robust cell sheet that we can harvest very simple with a magnet. And this is a very interesting strategy, maybe not to use by itself, but as an augmentation strategy combined with other 3D scaffolds for tendon regeneration. Another strategy that we have been pursuing is also to target specific mechanosensitive receptors via magnetic nanoparticles. And in this case, we have target activin A that has been assigned to the TGF-SMAD2/3 signaling cascade that has been reported as one of the possible mechanisms for tenogenic differentiation, using beads, magnetic beads functionalized with the respective antibody. And with these strategies, we were able to see an increase in the phosphorylation of SMAD2 with this strategy as compared, for instance, with the supplementation of activin A in the culture medium. And using these strategies, we were able to observe an upregulation of the tenogenic markers at the gene and protein level, showing that this could be a very specific and effective way of stimulating tenogenic differentiation of adipose-derived stem cells. Another work that actually was performed a couple of years ago, but that raised high hopes for this work, was the development of these membranes. They are based on a starch polycaprolactone blends, a polymer that we have studied extensively in our group, doped with magnetic particles. And in this case, besides the in vitro studies, we have implanted these in the rats and we stimulated them for some periods of time using a magnetotherapy device. And the most interesting aspect of this study was that actually using the magnetic membranes and stimulating them with the magnetic field, we observe some beneficial effects in the inflammatory response, namely the prevention of the increase of the capsule thickness and also the inhibition of the increase of mast cells infiltration. So with this, we wanted to understand better what could be the effect on tendon cells, and particularly in a disease context or in an inflammatory context. And for this, we have used a well-established model of inflammation that consisted on exposing the cells to IL-1 beta tendon cells in this case, and then we expose these cells to different conditions of the magnetic field. And what we observed very generally was that under specific conditions of the field, in this case at a low frequency and for millitesla, we observed downregulation of the pro-inflammatory genes that were assessed and the metalloproteinases, and an upregulation of the anti-inflammatory genes and the tissue inhibitor metalloproteinases, which shows that under specific conditions of the magnetic stimulus, we can actually modulate the tendon site response to induce inflammatory stresses, and that this provides an additional tool to achieve tendon regeneration therapies. We went then studying the same the same model, but in this case culturing the cells in the same magnetic membranes that I showed you previously, and exposing in this case to the optimized conditions of the magnetic field. And basically we observed the same behavior, that is a downregulation of the pro-inflammatory genes and an upregulation of the anti-inflammatory genes that were assessed, while at the same time the conditions that we study of the magnetic field did not or maintain the tendon phenotype of the tenocytes. We are currently actually also evaluating the response in this model to macrophages, which seemed also to respond very positively to the magnetic activated membranes. And actually our preliminary results show that when culturing these magnetic membranes and then the activation of a magnetic field, the macrophages are able to polarize into M2 phenotype, suggesting an immunotherapy potential of this technique. But as I mentioned earlier, for tendon tissue engineering, the scaffold material is very important, and besides being a biomechanical component, it's very important also that these structures mimic as much as possible the natural architecture of the tissue. And for this purpose, we have been focusing on different techniques, some fiber and biotextile techniques, and also on multi-biomaterial bioprinting. Today I'll focus just on this part of the work. And actually for this part of the work, we have been focusing on some nanotool box that is composed of these magnetic nanoparticles, but also these cellulose nanocrystals that are particles that are biocompatible, but they are able to enhance very, very significantly the mechanical properties, and on the use of these electrospoon nanofibers that can mimic somehow the natural ECM topography of the tendon. And actually these particles that I just mentioned can be decorated with the magnetic nanoparticles, and in the same particle we can obtain both functionalities, the mechanical reinforcement and the possibility of being magnetically actuated. So with this electrospin setup, we are able to obtain these continuous aligned nanofiber threads that then, using textile technologies, can be assembled into yarns, and these yarns can be assembled into 3D braided or woven scaffolds, mimicking with these structures the different architectures and the different units that compose the tendon tissue. So here you see an example of the morphology of these continuous aligned threads, and you see how they resemble the collagen fibrils and how similar they are to the ones that compose the native tissue, and how this can be assembled into 3D scaffolds using these braided and woven technologies, resemble what we call the tendon unit. Most importantly, with these structures and with the reinforcement of the nanocellulose nanocrystals, we can obtain structures that have mechanical characteristics that are in the range of the native tissue, and moreover that can actually mimic its quite unique mechanical behavior, including this toe region that is a hallmark of the tendon biomechanical behavior. More importantly, when we seeded cells into these materials, both tendon cells or adipose-derived stem cells, and without any biochemical simulation, we could observe a very similar pattern of the expression of the tendon markers, which demonstrates very clearly that not only these structures are able to maintain the tenogenic phenotype, but they also induce the tenogenic differentiation just by mimicking the topography and the mechanical properties of the structure and of the native tissue. But as I said, we want also to incorporate the possibility of magnetically actuated these materials. So we went on producing the same type of fibers, but in this case with the cellulose nanocrystals decorated with magnetic nanoparticles. And actually the type of fibers that we obtain are very similar. The mechanical behavior is also very similar, with some slight increase of the mechanical properties in this case. But moreover, also when we activated these fibers culture with the adipose-derived stem cells, we obtain even a more robust expression of the tenogenic markers. And here we wanted also to understand better what could be the mechanism responsible for this enhanced differentiation with the magnetic actuation. And we went to study the presence of YAPTAs that are actually transcriptional activators that are mostly cytoplasmatic, but when they're activated, they shuttle to the nucleus where they regulate gene expression. And here, as we see, we found higher expression of YAPTAs under magnetic actuation, which shows that, which is indicative of some tension in the cytoplasms, which lead us to think that there is some deformation induced by the magnetic nanoparticles, deform slightly the fiber that induces this deformation and tension in the cell cytoplasms that is responsible for the activation of these mechanotransduction pathways involved in the tenogenic differentiation of stem cells. Here, as you see, there is not only a significant increase of the tenogenic markers under magnetic stimulation, but also, again, a decrease of the pro-inflammatory genes and an increase of the anti-inflammatory genes that we have assessed, the markers that we have assessed in this case. The same behavior is observed when we move from the fibers that I just showed to the assembles of these into 3D structures. So the deposition a quite robust matrix expressing the typical markers of the tendon extracellular matrix, which can actually be enhanced when we, for instance, functionalize these fibers with tropoelastin, that is a protein that is also part of the tendon native matrix. And as you can see, with this functionalization, we obtain an even more robust deposition of the matrix. So with this, the main conclusions are that actually mimicking the hierarchical structure with the techniques that I show and with the magnetic actuation, we can only not trigger the tenogenic differentiation of stem cells, but also play a role in the modulation of the inflammatory response, which are key aspects to achieve better therapies that can regulate tendon regeneration instead of the tendon healing with the scar tissue. And with this, I end my presentation, thanking the founding bodies and also the team that has been involved in this work, and which is not working now. Okay, thank you very much.
Thank you very much for the presentation.
Thank you.
So the talk is open for discussion and questions. Any question from the audience? So there's one here, over here, left side. Wait a minute for the mic.
Thank you for the nice talk. There is a difference between acute ruptures of tendon and degenerative. Have you addressed that? And is your toolbox capable to sort of provide for both? solutions?
Thank you very much. No, we definitely need to address that, and we not only need a toolbox to address the different types of tendon, as I mentioned, but also those situations for sure. And I actually think that the difficult part of that is that sometimes we don't have the right diagnostic tools in order to better have a early diagnosis when we have some pathological conditions that usually drive sometimes to acute injuries. But definitely we have some of these strategies can be tuned and can be modulated to target different strategies for pathological conditions or for acute injuries, and that's our goal to address that in the future. There we have developed a wide platform, and now we need to study what can work better in each specific situation. But we need to target definitely better the pathologies that most of the times occur before more severe injuries that are perhaps more difficult to solve.
Thank you.
I have a question about the source of cells that you're using. So you mentioned amniotic stem cells. So the question was, do you have a bank, GMP grade bank that you have to use in clinical practice? And the second question is do you need the immunosuppressive therapy if you?
We don't have specifically, we obtain the samples with a protocol with the local hospitals, but there are already banks of adipose-derived stem cells, as these have been quite promising for a number of therapies, and because they are widely available and can be obtained in larger amount, there are already companies and banks that provide those, yes.
And you need to use the immunosuppressive therapy because it's an allogenic setting.
It's still I won't say it's totally consensual, but in fact, adipose-derived stem cells, they have shown to have an immunomodulatory behavior
effect.
quite similar to the bone marrow MSC. So in principle, yes, they should allow use in heterologous conditions.
Okay, thank you. There is one question.
So I'm wondering what's the implication of the YAPTAZ activation inside these constructs, given that normally what happens is that when you have the activation of this pathway, you have a fibrotic process. So for example, YAPTAZ is associated to activation of cancer fibroblasts. So can you tell us something more about what's the functional implication of this pathway activation into your constructs? Thank you.
Thank you. Well, it is a mechanism that has been involved. There are many mechanotransduction pathways that are actually involved in different processes. This is what we observed, but there are other mechanisms that we are exploring. I cannot really, I don't know if I can really answer your question, but we need to modulate very precisely to know what to target. But as I said, there are still a number of issues that we don't know about tendon development and what are the specific mechanisms that are involved. We are studying some possibilities, but we don't know exactly if this is the case. This was just an hypothesis that we have studied, but we need to address that more carefully for sure in the future.
Okay, so we will. There is one last question, very quick.
Yes, on the clinical setting, one of the most important problems is scarring rather than the lack of tendon tissue. Are you addressing this issue as well?
Yes, like I said, that is, we really think that is the critical issue in tendon regeneration for sure, and that's why we need we think that the key point would be how we can modulate the inflammatory response, and especially in very early beginning. And that's why we are using this technique that apparently, and from our results, it seems that can provide us a tool to address that. But we need to address that very carefully, of course, and know exactly what to target and which stage and how we can actually use this tool successfully. But for sure that this is the most important issue, to be able to shift the process, the remodeling process, in order to achieve a functional tissue instead of scar tissue, for sure. That is the main problem.
Okay, thank you very much.
Thank you.
So we have to move to next talk. Will be my co-chairman, Dr. Uwe Marx, is the CEO of Tissues Germany. The talk of Uwe will be from organ-on-a-chip tools towards patient on chips, enforcing a paradigm shift in drug development.
Thank you very much. So what I want to introduce to you is the basic problem of drug development. One of the drug development dilemmas is a really low predictive power of preclinical testing, right? Only 2 out of 10 drugs which have fully run through the preclinics make it to the market due to a high attrition rate here. Now, and this is a value inflection point or disruptive idea 15 years ago. The community came up, the in vitro community came up and said, okay, if we can really grow up human in vitro cultures toward systemic complexity, making a patient on a chip here, we then probably will be able to disrupt or to make a value inflection in this area. And this value inflection has been estimated by the stakeholder community in 2016 to, if that comes true, to have an impact and significant impact on time and economics of drug development, making the costs 5-fold lower and the time 2-fold lower. And if you expect that this can become true, you know which kind of power might be within that background here. Now, with regard to the history in science, there were a few laboratories starting already at the beginning of the century as pioneers in the 2 development areas of single organ engineering, which was MIT and the Wyss Institute at Harvard, and the Cornell University and our university here, the Technische University, where tissues is a spin-off of in the area of multi-organ engineering. Now, over the time, this area has really revolutionized due to a large American program. And even in the multi-organ areas, we have fancy developments on a scientific level, on a scientific method level, as the Physiome program here, or the menstrual cycle of women on a chip here, and other things. Now, in 2019, after having one first science paper in 2006, the one of Don Ingber for breathing lung on a chip, in 2019 is again a remarkable year, because again, organs on a chip made it to the science and were combined with organoid ideas, which came across in vitro cell culture, right? Now, in this year, we are expecting more than 1,000 publications. We had them already last year, and we have counted approximately 360 research groups in the field. Now, how can you run these machinery into a really industrial application? For that particular question, the stakeholders are meeting already a second time here in Berlin in June this year. So the first stakeholder meeting was in Berlin, and this is, we are proud to host them here, and the second was here. And this report has been completed last Friday. So I'm happy to give you a number of data here. There are still some FDA, Chinese FDA, EMEA clearance processes. So the submission of the report for this workshop will be delayed by this clearance. Some pharma companies need to clear the report, but I can already give you a number of findings out of this report. So the important thing is what we found out, and what we could see is that along these 15 years, these technologies have advanced from purely scientific backgrounds, with an European society being founded last year, into spin-offs and supplier industries, where at least 4 standardized commercial platforms have appeared over the last few years, and more than 28 companies are around here, through feasibilities into adoption for pharma with fit-for-purpose assays, and we are trying to go into regulatory acceptance. And the first paper from the regulatory or FDA point of view appeared recently this year again in literature. Now, what was the countable effects of these 15-year developments? The countable effect of making validated assays out of standardized commercial platforms is 23. As of Friday last week, we counted 23 data sets or assays which went into end users' portfolio decision making. And the next step, context of use assays, are unfortunately still 0 until Friday last week. And we think that the first #1 data set coming from an MPS-based technology flowing into the IMD or IMPD will come within the next half year, but still a way to go. So it took us 15 years here. These are the 4 platforms. And the interesting thing here for the commercial platforms is that indeed the platforms appeared on the basis of the pioneering groups. So this is with Harvard, a spin-off in the US. These CN Bio in-licensed the Grinder Liffes Labs, the MIT technology. This is Technical University, and this is University of Leiden. So all of them have 15 years of knowledge how to develop the technologies. That makes it quite a strong message. Now here is this 23 cases table, still unpublished. And I just selected one of them to give you a feeling of what these validated assays are in contrast to are about. So all of the technologies somehow try to reduce the human organs into a smaller downsize. Our technology at tissues is downsized by a factor of 100,000. So each of the organs is 100,000-fold smaller than the original one. And we try to combine different organs or run single organs in the right ratio with each other in a common multi-organ vasculature. The example I wanted to show you here is a so-called bone marrow, single bone marrow assay. So the bone marrow unit is an hematopoietic stem cell niche unit of 300 micrometer here. We are mimicking this in a ceramic fashion here in that microfluidic system, and are using mesenchymal stromal cells and hematopoietic stem cells, which are seeded first mesenchymal stromal cells in blue here in that matrix, and then human hematopoietic stem cells in a right ratio. And then you can see that they grow within that 3-dimensional spongious bone-like matrix. And over the time now, over 4 weeks or even longer, you can harvest cells and always can show that we are able to show that you can get all the colony-forming units and you can repopulate the cells harvested from these kind of hematopoietic stem cell niche into mice or into the geles and can generate all human lineages. Now that stimulated the industries to come up and say, let's qualify and validate this kind of assays, right? This was just scientific papers. And then 2 years ago, we started with the AstraZeneca safety unit, which was aiming to make repeated dose hematopoietic lineage toxicity, which is not very predictive in rat and mice for humans, to make it an assay for this prediction. And we immediately were forced to prolong the culture period to 8 weeks. We were forced to make self-supply secured, reduce the costs, validate the assays, control resident and circulating cells, so the circulating cells, which are the mature cells, and the residating, which are generating the new ones, and make an inter-laboratory long-term qualification between the laboratories. Then the assay was established at AstraZeneca. It immediately spread to any of the other pharma companies. We are step by step introducing that assay into the scene. That should serve as a role model for you for a single organ human bone marrow toxicity assay, which made it to industrial adoption. Now, in advanced biotherapies, cell therapies, human cell therapies, they do have animal models behind, but all of you know that even humanizing the mice, they are still phylogenetic distant from humans. And that's why using organ on a chip or multi-organ chip technologies are relevant. Now, in this case, we are not talking about a single organ. We are not talking about just a single combination, but we are talking about the combination of multiple organs in a vasculature with identical organs being autologous in order to prevent transplant or rejection. And we were able to make a 4-organ autologous chip recently and publish that. And the number of groups are now running this into universal physiological templates. We think that an organism needs at least 10 organs to be combined to stay stably homostatically and then to induce disease in that models. And I invite you to read the respective references. Now, the program here at Restore, you have seen that 3 of the 4 platforms which have been established are European ones. So we are strong with preclinical models here in Europe. The program here is quite large. I don't want to go due to time restrictions to throw, but we need to emulate organismal homeostasis on a body level. We need to establish this chronic disease model and the respective assay format, maybe even longer time than two months. And we need to run it into automation and data management. So those are the basic aims here. Our next large event, which summarizes anything you want to know about organs on a chip in this world, and there is a lot, is the 11th World Congress on Alternatives to Animal Use in August in Maastricht. I invite you to join this, to learn about these different aspects of the technology. And finally, I would like to thank our joint team of the university and Tishis to running many of these assets and bring them into hopefully regulatory acceptance, finally making them ICH guidelines and replacing the animal guidelines. Thank you.
Thank you very much, Uwe, for this talk. Any questions from the audience? One question there.
You briefly touched upon this, but can you comment a little bit more about perhaps mimicking disease states using this technology? So I thought when I saw your bone marrow model to think about residual disease and hematological malignancies and so on and so forth.
Yeah. Very good question. We currently have 23 single organ different models. Many of them have reached the disease stage. One of the front runners is Don Ingber's lab at the Wyss Institute. So we have lung fibrosis, we have BDI in the intestinal models. We are working with AstraZeneca on a diabetes model, combining a liver and a pancreatic islet background. And we have a number of oncological backgrounds. So you can use the bone marrow, for example. We developed a combination of a healthy skin and a tumor for Bayer to make it a safe-efficacy, let's say, safety and efficacy test system with a healthy organ and a tumor. There is a lot there. And the American program produced 502 papers only on trying to integrate disease into a single organ model. Still, the combination and immuno-oncology or immune-based systems are not there. We need vascularization, immunocompetence, and innervation in these systems. This is some of the challenges, but I invite you to read the report, which probably after peer review will come up in the first quarter or second quarter of the next year, and will make all these things and all the roadblocks and a 15-year roadmap available.
One more question there. Back.
How far is it with integrating the microbiome into your chips?
a number of chips, for example, the emulate background is already able to host microbiomes on an villus-like two-side perfused intestinal part. So microbiome has been integrated in organ-on-a-chip models, primarily in the intestinal models, but some of the people which are doing skin and hair on a chip started already with microbiome, and there is a little bit about lung and pathogenic or lung microbiome, which is not such a large thing like an intestine. So it's coming up.
Any other questions? I have one question actually.
Okay.
Can you, because there's no standard in the human bone marrow on a chip. It is quite very difficult. Actually, nobody has really done there are some classical papers with transplanting human ossicles, into NOG mice, immunodeficient mice, and then you inject human cells to see if. What would you use this kind of model as a comparative to your human bone marrow on a chip? How would you compare to?
They are strikingly good systems in making it a human bone marrow on like a sponge-based background, and then using the mouse like an incubator and making vasculature. These are the best comparators to making our bone marrow vascularized now. And we will use these ones again. It's always a hybrid system. There's the backgrounds of a hybrid system.
Okay, thank you very much for Uwe. And we are now moving to the last talk of the session by Emmanuel Donadieu from INSERM France. Emmanuel Donadieu is working in Cochin Hospital. Is interested in essentially capacity of T cells to migrate and recognize foreign antigen, immunology field. And his talk will be entitled Monitoring CAR T cell efficacy in fresh human solid tumor slices.
Thank you very much. Thank you for the kind introduction, and I'd like to thank the organizers for inviting me to speak today. So indeed, I'd like to actually continue the discussion that was started by the prior speaker and present an innovative preclinical model that enabled us to visualize T cells, especially CAR T cells, and test their efficacy in tumors, including human tumors. So as we've heard this morning, CAR T cells can be very effective in certain type of B-cell malignancies, as shown on the left, especially acute lymphoblastic leukemia or lymphoma, but not yet against solid tumors on the right. And one important challenge in the field is really try to understand why CAR T cells are not effective in controlling the growth of solid tumors. So for CAR T cells to perform their job to control a solid tumor, they need to take several steps, which are summarized on that scheme. They need first to enter into the tumor, extravasate, then navigate within the tumor microenvironment, make contact with stromal cells, stromal component, reach the malignant cells, produce their cytotoxic activity, and then remain and persist within the tumor microenvironment. And we know now that all of these steps are controlled by both intrinsic but also extrinsic determinants, biomarkers, that could either stimulate or inhibit the functioning of CAR T cells. And I've tried to list some of the determinants that have been identified so far, but you could see that there's still a lot to learn about these factors that control the behavior of CAR T cells. So that provokes, obviously, a number of questions. Two of them are listed here. How to identify biomarkers that would predict sensitivity to CAR T cells. And the next question, which is actually related to the previous one, is in which model can we test and predict the efficacy of CAR T cells? So as we've heard before, a number of models, preclinical models, have been developed. All of these have a number of advantages, but also limitations. We've heard about PDX, organoids. For example, organoids, they most of the time lack the tumor stroma organization, and also most of the time they're also devoid of the immune system. So we are at INSERM in Paris, we have developed a different method, a complementary method, an organotypic model, which is based on human tumor slices. So the workflow is summarized on that scheme. So in brief, we have ongoing collaboration with clinicians working in Parisian hospital who provide us with fresh fragments of human tumors, lung, renal, ovarian. And the first step is the tumor is collected, then embedded in agarose. We then cut the tumor with a vibratome. We usually perform 300 to 400 micron slices. And on top of the slices, fresh slices, and nothing is fixed, obviously, during the whole procedure, are plated some T cells, could be T cells purified for the same tumor, the T, so it could be genetically engineered T lymphocytes, so that CAR T cells. The slices are placed at 37 degrees for half an hour. Then slices are rinsed to get rid of the cells that have not entered into the tissue, and then systematically immunostained to label stromal cells, stromal component, but for cancer cells. And finally, we used real-time dynamic imaging microscopy, either confocal or 2 photons, to look at the behavior and the responsiveness of CAR T cells in relation to the tumor microenvironment. And so we've been able, in this condition, to actually assess the distribution and migration of CAR T cells in a preserved human tumor environment. I'll show you just some example. For the EGFR CAR T cells are in green, the stroma is in red, and the tumor cells are in white. And so you could see that the T cells migrate very actively. And so we have also developed automatic tools actually to extract data from these movie, for example, to analyze the velocity of T lymphocyte CAR T cells in such a condition. We also looked at the activation of CAR T cells, focusing on the calcium. The calcium is a second messenger that rapidly increases when T cells get activated, and we use a number of dyes like Fluora2 or Fluoro4 actually to look at the calcium response against one example of CAR T cells. And you could see that the CAR T cells, the calcium is colored in pseudocolor, and the CAR T cells rapidly get activated when they interact with their target. And again, we have developed a number of tools to very precisely quantify the amount of calcium in CAR T cells at a single cell level. Several hours after exposing the tissue, the fresh tissue, to CAR T cells, we have also been able to look at the cytotoxicity, the ability of CAR T cells to kill malignant cells using caspase-3, caspase-7 fluorescent dye, and to show that in some condition, the CAR T cells were able to infiltrate tumor islets and to show potent tumor killing activities as evidenced by an enhanced apoptotic cells or green fluorescence. We have also, in collaboration with Milteni, we have also combined the slice assay to the Maxima technology, which is a ultra-high content imaging platform, which is based on the repetitive staining and erasing of fluorophores or fluorochromes, and which enabled us to stain a cryosection with several tens of directly coupled antibodies and really try to map the tumor microenvironment in relation to CAR T cells. This is just an example. And with these tools, we have been able to identify a number of obstacles that prevent CAR T cells to perform their job, especially to migrate and reach cancer cells. The first obstacles that we found is the extracellular matrix. You may know that when a tumor grows, it gets stiffer, and we found that the fibrosis going on in tumors, in human lung tumors, has a detrimental impact on T lymphocytes, preventing them to reach cancer cells. This is an example of fibronectin fibers surrounding tumor islet. The tumor cells are in blue. You could see that the T cells are quite far from tumor cells. Or when they are present close to tumor cells, they migrate along the fiber, also sequestering T cells in the stroma. More recently, we found that macrophages, which are very abundant cells within the tumor microenvironment and which have a very well pro-tumoral role, have also a negative impact on T cells, impeding them from reaching cancer cells. So I'll just show you two movies. On the left, the control condition. You have the tumor cells in blue, the CD8 T cells in red, the macrophages in green, many macrophages in the stroma of tumors. So you could see that during a 20 minutes recording, most of the T lymphocytes are quite stuck into the stroma, forming long-lived interaction with macrophages. Now, when we remove macrophages and we design a protocol to get rid of the macrophages, and so no more macrophages on that settings, and now look at what's happened to T cells. So you could see that the T cells, they migrate very actively, also more prone to contact their target. And in mouse tumor models, we have found that this macrophage depletion strategy increases the efficacy of immune checkpoint inhibitors. So in conclusion, we have developed a new preclinical assay to monitor T cells efficacy, including CAR T cells in human tumors. This system presents a number of advantages: a preserved tumor organization, a short generation time, a good access to the content of the tissue, and as you saw, it is amenable to imaging microscopy. Of course, like any system, it has a number of limitations with a possible loss of important component during the slicing procedure, and also a relatively difficulty to maintain the system several days in cultures as compared, for example, to organoid. But combined with real-time dynamic imaging, microscopy, and automatic analysis tools, we have been able to identify a number of biomarkers, and we are still working on novel biomarkers, and to determine also the mechanism of action of CAR T cells in human tumors. So the next steps would be to try to push the system and in order to monitor later CAR T cell responses, such as proliferation and persistence, which are very important step in the efficacy of CAR T cells. Try to really be patient-specific, try to work with CAR T cells and tumors from the same patient and try to see whether the system could predict the efficacy of CAR T cells in such a setting. Also we've heard that toxicity is a very important concern in CAR T cells, so try to predict CAR T cell toxicity in slices from healthy tissues, and also try to combine the slice assay with the organ-on-a-chip model through the RESTORE program. So our lab is located in Paris, a cancer and immune response team, an INSERM team, and I'd like to acknowledge all of our collaborators, including clinicians who provide us with fresh fragments of tumors, and all of our colleagues from the CARAT consortium. Thank you very much for your attention.
Thank you very much for this talk. I'm sure there are many questions. I have a question about, you mentioned this, but in terms of persistence, detection of persistence, so you need monitoring the assay, during clinical assay. So could you also, for instance, detect exhaustion during your technique?
Yes, I mean, it's a challenge because, as I said, the slice assay, organotypic slices, it's relatively difficult to maintain the system over 2-3 days or 1 week. For early responses that I showed, that's fine. Even for cytotoxicity, that's okay. So, of course, we plan to look at proliferation, probably day 4, day 3, day 5, probably persistent exhaustion. Well, maybe exhaustion, to map exhaustion is probably a number of markers. We could look at PD-1, we could look at TIM3 and other markers, which probably you don't need the slice assay for that. Yeah.
Well, I take advantage. I have another question about there are some tumors, of course, which are, you know that they have targets like glioblastoma, but the CAR T cells are notorious, you know, they don't go into the brain. In your system, would you, for instance, be able to see if from the diagnostic biopsy, you could predict if, let's say, CAR T cells directed against EGFR receptor in glioblastoma would be a good target?
That's exactly the goal. So far, this system has been instrumental to identify a number of biomarkers. But now we would really like to predict the efficacy of CAR T cells using CAR T cells and the tumors from a single patient that received or not CAR T cells, and to know whether the system is predictive enough, strong enough to predict the responsiveness of CAR T cells. Absolutely, yes.
Okay.
Thank you.
Thank you. There's one question.
Yeah, thank you for the talk. Did you also had a look in the importance of the endothelial cells or the angiogenic properties of your tissue and your T cell interaction?
Well, so far, just to remind you that the slice say we add the CAR T cells or the T cell onto the section. So in a way, we bypass the physiological entry of T cells into the tumor. These steps we totally ignored in that system. But we did find the fact that when we add T lymphocyte onto the tumor, some T lymphocytes were located close to the vessel and actually migrating close to the vessel, which might suggest that you have a specific environment enriched maybe in chemokines close to the vessel, which is favorable for T cells to migrate along the vessels.
Okay, Uwe, any questions? Okay, thank you very much. So I think now this is the photo session. I think this is the photo session. This was I here on the. Okay, thank you.